So, and sometimes we do other things, we talk about some other stuff as well. So use those lecture slides as a guide, like if you want to review what we talked about. But also, so you don't have to write down every little thing that's on a lecture slide. Take notes about, general things what we're talking about, things that I'm talking about, but don't feel like you have to be like, oh, can you go back? Can you go back? You don't have to ask that because you can access most of those slides will be accessible for you, okay? Also, I was asked during the break about just worry about having enough time to study and do the work involved with this class. And maybe I might have exaggerated a bit about how much time you need to be able to take outside this class. So I would say if you could think about the amount of time it might take for you to finish up your labs, reading the chapter, doing a couple, maybe a video, doing a couple different assignments. You're looking at probably at least like 4 hours a week. I know that doesn't seem as much as what I said. I did exaggerate a bit, but it just depends on you and how much background you have in general biology before coming in here. If you've already taken zoology here and Bio 1 or general Bio 10, you're going to have an easier time in this class. If you haven't taken a biology class in years, some stuff that I go over pretty quick, you're going to feel kind of lost on, and you're going to have to take that extra time. So if I say between like 4 hours and 10 hours that you might be having to put in to truly do well in the course, and maybe 1 week you put in 1 hour, maybe the next week you put in 5 hours or 6 hours. You know what I mean? So it's going to be totally up to you how much time you put into this class outside of class. Okay. I just wanted to address that because I didn't want to scare you and freak you out. This is going to be a challenging class, but it's definitely doable if you put the time in. So, what makes a plant a plant? Yes?
There's like photosynthesis and like cool stuff.
Yeah, so photosynthesizing, are there other organisms that photosynthesize though?
Mm-hmm.
Yeah.
It's a eukaryotic organism that photosynthesizes.
All right, cool. So eukaryotic organism. Anybody want to tell me what a eukaryotic organism is? Yes?
Multicellular organism.
Well, many of them are multicellular. Sometimes eukaryotes can be unicellular, but you're going in the right direction there. Multicellular organisms are definitely eukaryotic, for sure. Yeah, anything else I want to add?
A eukaryote is a cell that has a true membrane and true nucleus and membrane-bound organelles.
Awesome, yeah, and then, so nucleus being membranous structure that surrounds the DNA, or the nucleus, right? Very good, and yeah, so then the organelles inside membrane-bound organelles. Bacteria, archaea, they're not considered eukaryotic. So, right, plants are all eukaryotic, so awesome. They evolved to photosynthesize, but we have a few examples in the group that don't photosynthesize. They've lost it, and they depend on other plants for their food. Yes?
Cell walls.
Cell walls, awesome. They do have cell walls. Some other organisms that are not kingdom plantae do too, but kingdom plantae, there's a certain component that the cell walls are made of. Anyone know? Cellulose. So we're going to be getting into one of our topics. We're going to be jumping into more details Thursday. Carbohydrates. So cellulose falls into that biological molecule. So cellulose is a major component of cell walls of plants. So some other organisms have cell walls too, but they may be composed of different things. So mushrooms, so fungi, their cell walls are made up of a different type of carbohydrate. Anyone remember what that's chitin, which is also found where? Anybody who took like zoology recently? Yeah?
Insect wings.
Yeah, insect like exoskeletons. So, and that's one of those interesting links linking fungi and kingdom Animalia together is the DNA, what the DNA tells us, but then also they have that interesting similar molecule. Okay. So yeah, if fungi do they digest and break down externally what's around them, and they break down plant material, right? They're decomposers. And if they were to break down, they would potentially break down themselves if they were made out of the same material, cellulose. So, anything else? These are good points. Photosynthesizing, yeah, but doesn't it seem like I'm always like, yeah, and yes, that's true, but, and that's just the study of life in general, biology. There's just exceptions all over the place, right? So we try to like put things in a neat little box, and it doesn't really work very well. Anything else you want to say about plants?
They're pretty cool.
They are very cool. I like that. Yes.
Lack of mobility, like they're not able to move around.
Yeah, that is definitely something that can separate plants apart from other things. Anybody have any thoughts on that? Any exceptions? But yeah.
Tumbleweeds.
They don't like I don't think it like actually moves on the ground itself, but isn't there like a walking onion that like bends over and sprouts?
There are some really interesting plants that do that. Like there's some different plants that will like practically walk in very slowly with growth. And we see plants move with the sun. There's definite movement. Plants have movement. So that's where the but really is yes, plants don't move in the way we think of when we think of animals or certain kinds of protists. They do move. And you guys will see like some really cool cells that are called, sperm cells, right? Well, plants have sperm cells too. And some plants actually have sperm cells that are flagellated. And they will swim, they move. And so that kind of like, you're like, wait, that's a plant cell. How's it moving around? And so mosses and ferns, they've got to have water in order for the sperm to be up an egg because they're flagellated sperm. Very similarly to animals. Actually, way cooler though. Their sperm, instead of just one flagella, they have like multiple flagella, and they'll like spin around, and they look really cool. So we'll see those up close. And yeah, so just the movement plants do as they grow, and then moving by way of their seeds, plants can get all over the place and spread around. I talked about one of the things I looked at with my dissertation was how this particular plant family started in, I think, South America and how it spread out throughout the world. So, all right, any other things you want to throw in? Plants being immobile is one of the, and cellulose and having some other things we're going to talk about is one of the things that links them to us being able to use them so well. We don't have to, run after something. We know where this plant is, and we can go there throughout the year to get food or medicine or whatnot. All right, so I think you guys hit it pretty well. And shoot, can you guys put your name tags up? So it helps me remember your name. Now I have it set up that way. And what's your name? Rita. Yes. Okay. So Rita brought up a good, cool point on multicellular aspect. Plants are definitely multicellular. And then eukaryotic, photosynthetic. This is kind of like not something we really talked about, but we'll talk about this more. Producing embryos within gametangia. So there's lots of organisms out there that they'll just like throw their sperm and eggs out. There's some animals, very simple animals that do this, sponges. out in the ocean. They'll send out the eggs and the sperm, and then hopefully they connect together. And what's that? Sea anemones, and a lot of algae will do this as well. They'll send out there, and so hopefully they'll connect together. But plants, they keep the egg inside, and so the sperm has to get inside where the egg is stored, and fertilization takes place inside, and that embryo is inside of a special, almost like a womb for that plant, and from the mother plant, and is protected and cared for, and given nutrients. And then depending on if it's a seed plant, it goes dormant and is encased in extra protective material of a seed coat. So embryos stay within gametangia. They contain chloroplasts that of course connects back to photosynthesis and cellulose cell walls, lacking power of locomotion. So you guys did awesome. You pretty much hit just about everything. So good job. And so why study plants? Yeah, it was brought up that they're cool. Yes, that is why we are studying. That's why we're here. They're cool. They are amazing. Anybody know what plant this is?
Yeah. I
What's
don't
it?
know the name, but I know.
What's that? Sundew. Yeah. So I heard a couple of you said Venus flytraps, that you like Venus flytraps. This is Venus flytraps. This is not a Venus flytrap, but it is also a carnivorous plant in the way that it catches insects like a Venus flytrap does, but more like kind of like fly paper. If you think about it, like these are hairs, and they have a sticky gland on the tip. And this is actually a modified leaf, as you're going to get to know different plant organs. And the insect is attracted and gets stuck here. There's some yummy, tasty material that's produced on these little sticky structures. And they're sticky. And then it can fold over onto the insect, and it doesn't absorb or dissolve the exoskeleton, the chitin, and but it takes what it can from the inside, and one of the major components that it's trying to take is the nitrogen. So these will normally grow in areas where it's lacking a good source of nitrogen in the soil. Plants, this continues along the lines of why plants are amazing. Not only are there really interesting carnivorous examples, but there's some really cool examples of coevolution with their animal pollinators and orchids. The orchid family is one of the largest plant families in the world, and very unique, and they really have learned how to take advantage of their pollinators to the point where usually you think of pollination, plants giving something, and in return it's getting pollinated. Well, sometimes the orchids trick the insect into thinking it's going to get something like sex, and it doesn't get anything in return. So there's some orchids out there that look and smell like the chemical signal is matched. Look and smell like a female insect ready to breed, and the male comes over, tries to take that female insect, which is really the flower, and in so doing, pollinating the flower. So pretty interesting and tricky. So we'll be going through some of those examples throughout the semester. Plants are, some folks might, have some different ideas about this, but generally, plants are the largest and tallest organisms living on Earth. Anybody want to challenge that? What about the oldest? You brought up jellyfish. Anything interesting about the age of jellyfish? Have you heard? Yeah. Did you bring up jellyfish?
I didn't, but I know that there are.
Oh, I thought you.
jellyfish.
Were you saying jellyfish with like the sending the eggs and the sperm out?
No, sea anemones.
Oh, sea anemones. Okay. Well, they're in the same. They're in a similar group here. But yeah, my brain just thought of like areas. Huh? What's that?
And
Oh, anybody in back? I thought I saw somebody. Yes.
For the oldest, it's sharks.
Sharks.
Sharks are older than trees.
According to who?
Lean.
Oh, is that what is Randy saying this? He's spreading this around. Randy's a good friend of mine. I'll chat with him about that. But there are some interesting things. So jellyfish it's thought they'll go back and forth from their immature to their mature stage and can do this really interesting thing. Isn't there a movie like that? The Mr. Button or Benjamin Button? Isn't that like he gets young again? Well, there's some animals that are thought to do that interesting back and forth thing. But yeah, so this oldest actual individual tree, because there are some trees out there that pop up new ones underground. And there's some fungi that are also huge. You guys heard of the humongous fungus?
Mm-hmm.
And well, sharks can get pretty old. I think there's still some question about exactly aging them is tricky. But with this for instance, the bristlecone pine, you can age that because you can count the tree rings, so you know for sure how old that individual is, and they've been aged to over 5,000 years old. Yes?
Isn't that like oldest living organism title also partly just because they're the only ones with the proof of the age?
Yeah, and that's why we're having this discussion, because it's like, well, yeah, we can actually count the age of these trees. And we don't have to cut them down. We can do a core sample of them. But whereas some of these others in Kingdom Animalia, it's more challenging to get an actual age. But
And are those the ones in Joshua Tree National Park? Or-
no, these ones are in the White Mountains on the east side of the Sierra Nevadas.
Okay.
So oldest and some of the largest organisms. All right, so lots of reasons why plants are cool. Photosynthesizers in general are amazing, and we should tell them thank you every day. Pretty much every time we breathe, we should be thanking the photosynthesizers in our life. And some people will say, and as any of you guys who are interested in marine biology, there's so many organisms out in the ocean that are photosynthesizing that every time we breathe, we should thank the ocean photosynthesizers for one breath, and then the next breath, the terrestrial. And thinking a lot of the rainforest as the lungs of the earth as well. So both aquatic and terrestrial environments. And cyanobacteria, not just plants and algae. So if we look at different terrestrial environments on Earth, we see plants in many different species, different forms, and of course when you think of ecology, the photosynthesizers are at the start. I always like to say the bottom. That almost makes it seem like they're not as important, right? But they're at the start of those food chains. This one looks familiar, right? California. This is definitely a California oak. And of course the ethnobotany side of it. Many different uses. We depend upon plants for much more than just our oxygen, but food, clothing, construction, medicine, and so forth. All right, let's go ahead and grab our microscopes that you're going to be using for the semester. And we'll see if we need any extras from the other room. So let me just show you where these are microscopes.
If we drop them, do we have to take them?
Yes, they are pretty expensive. Be careful. Don't drop. You'd have to work out some. You have to. That's what happened. That's why Sarah's been working here for so long. Drop the microscope.
Oh, yeah, she's paying it off.
She's paying it off. All right. So
Worst nightmare.
there's a little handle here. They are kind of heavy. So if you're taller and you can reach easier, go for the the taller ones, just to help those of us who are a little not as tall be able to easily get our microscopes out. What I want you to do is, and they should hopefully be in the right places, maybe they're not. When you grab your mic, yep, that one is. You'll notice there's a little tape here with a number, and then there's a number here on right below the spot that they should sit in. So you're going to grab the microscope and get it out, and you're going to start getting it set up. Okay? So go ahead and I'll take it just a little bit just to everybody grab the microscope and come back to your seat. And then let me know if you need any help with anything. The plug sockets, they're by your knees. See that there? And that's your plug socket.
Right here.
Box number 10. Oh, grab it faster than you. Usually per person, we have 36 people.
Yeah, well, she was if we run out, we'll grab some from the other class. Yeah, we're still in yeah, just like chairs.
Will we ever be using the gas?
Not in this class. They are actually shut off, so we actually bump it. We don't do
Say that again?
The main gas supply valve is shut off.
Okay.
So you can't max
I'm
it.
so excited for you guys.
Yeah, there's the socket like right next to you.
Literally right here. Oh, as in like there's something I pull out. Yeah. Big dial on the bottom.
There's one for course movement and fine right here.
It feels like it's not plugged in properly.
Oh, that's pretty funny. Did you?
So it's focusing.
Why does it do that?
I don't know, but it is okay. So it's probably just an
Alright,
indication
so
of
you're
light level.
getting a lab. This lab is gonna be what we're gonna be working on Thursday as well. Okay. So this is a microscope / biological molecules lab. So we're gonna be focusing today on getting to know your microscopes and then flowing into Thursday biological molecules. Okay. So we're definitely not gonna be getting. We're only getting through the first few pages of this today.
That's the original.
Your microscope number is 4. I have no idea. Don't. Okay. Why is it 4? Was mine? Yeah, mine was 23.
Oh, yours was just wrong.
It was just in a completely different area.
Yours just probably belongs in the other case then. That's unfortunate.
it's fine, that's what it is. That just means I put it back in the right place.
Yeah, I can't see their like.
Let's just see how many books.
No on off switch.
All right, everybody.
We'll just throw it
If you feel like they're not stained well, or you feel like they're like kind of washed out, you can always try to go and grab another.
I think I tried to make sure those are all good, like this.
Yeah, it might still be a good one. You could try it if you want to. But you can also just have them here.
All right, how many people feel really comfortable with the microscope already? All right, cool. A few of you do.
Present?
Awesome. But a lot of you
Oh, that
may
one's got a
not.
longitudinal
So
section
that's
too.
kind of the goal of today's lab here. Get comfortable with microscopes and practice making a few slides as well. All right, so Sarah's testing out a slide of a stem cross-section of corn. I was trying to remember what we're looking at. So it says it's Zea mays corn. So that's what you're looking at. And go ahead and take your lab, the first page of your lab has an image of a microscope that looks a lot like yours. It's not exactly the same, but it's pretty similar. And I want you to get familiar with the different components. And on your lab too, you have all these microscope parts. And I forgot to number them. I forgot to put the numbers. Anyways, this gives what those parts do, and actually not even all the labeled there. I actually updated this with a new picture, and I forgot to add the numbers, to be honest. That's why the numbers. Sorry about that. But let's make sure everybody look up here, and you've got your microscope in front of you. These obviously are the eyepieces, and you'll notice right off the bat that, oh, how do we do in numbers of microscopes? Do we have enough for everybody? Perfect. Wow.
Just enough.
Yeah, I think we got a couple more 2 years ago. So I think we're good. Perfect. All right, so can I borrow yours? I don't have an extra to make my sample on. Eyepieces. Remember I talked to you about your seat control. That's good to know about for your scope if you want to be sitting up higher or lower. Also, you can change these around to your lenses. You're going to be using these for 3 hours, sometimes at a time, and I want you to feel comfortable. I want you to be comfortable using both your eyes. If you have 2 eyes, let's see if you try to use them. I think everybody in here has both their eyes. If you didn't, it'd be fine. It'd work fine. But yeah, let's use our eyes, both of them. And these can sort of adjust a little bit. These can actually be pulled out so don't accidentally have it fling somewhere. And you can adjust them a little, but for now, just make sure that they feel good on your face. There you go. And also notice that it has a magnification on the little writing on the side of it.
We kind of ran out of slides. Can we give people in the back some of these for?
Yeah, that's what I had. Yeah, let's go ahead and adjust it. Just often I'll see the same thing. And tell them to make sure they look at this. Yeah.
Okay.
Cool.
There was actually some that had
That were bad?
longitudinal slide that.
Oh, did they?
This one might be bad. I'm kind of suspicious of this one.
Okay, so just hand up the Alright, so slides are coming to you, and I haven't really we haven't discussed how to put them on your scope quite yet, because it can be a little tricky. And, so, Kaylee, yeah, can you maybe walk around and just kind of help make sure they are putting their slides in right? You have your stage, and you have your little stage clip. You want to make sure you put your slide in there in a way so that it comes right into the corner, this corner. And pull your little stage clip out and set your slide in there. Like that. And then it's there. And then you'll be able to do access control.
Especially when you have like a thing. So I did get it to the
Yeah, exactly. Yeah. Yeah.
I have perfect. I can't see it.
Yeah. So just use one eye.
Just one
And
eye.
so if you want to, thanks for letting me know. Because I do often have students who they're squinting.
I
the whole
just closed
time.
this eye. I literally can't see anything.
Yeah.
outside.
Okay.
I just want to be magnificent and use both eyes.
Yeah, try to, because like what I'm saying is a lot of folks who can use both their eyes, they only use one
Yeah.
because they don't want to mess with getting it right, or it's actually really challenging.
I also haven't tried using. I didn't bring my glasses today. If I had my glasses, I'd be able to put my glasses before.
Sometimes wearing glasses is tricky. Sometimes people don't like to wear glasses when they're using it, so it's totally up to you. But yeah, so figure out what is a comfortable way to do it. You can even get this out of your way maybe more. I don't know.
I've used microscopes plenty of times, so
Yeah.
pretty.
And original microscopes are just single, unicular.
Binocular.
Yeah, unicular or monocular.
Monocular. Yeah, that's the word I was thinking of. I'm like, unicular sounds funny.
Like
Monocular.
monocule.
Yep.
It's kind of hard to your eyes straight.
Yeah. So when I say try to use both your eyes, really try to, but it is challenging. It's challenging, and sometimes you'll notice that one ocular is maybe pushed out further than the other. All right.
Honestly, the only difference between
So the next thing I want to point out
so you're not missing out.
is
I used it before. I couldn't see.
so has everybody put their slide in yet? So look up here really quick. Here's this here. This is everybody, let's just take a quick break and look up here. I want to point out a couple things. So Deanna and Sarah too. Let's focus up here just really quick, and then we can keep helping. But this is the stage, and these right here are the stage controls. And if you don't get your slide in here properly, then you're not going to be able to use the stage controls to move your slide around, and it's going to be really tricky. So really try to make sure you get that slide in there properly. And then these are the nose piece objectives. I just call them the objective lenses, and this is the nose of the microscope. Make sure you're starting with the lowest magnification possible. So lowest magnification, which one do you think that is?
4.
What color is it?
Red.
Red, and what's the magnification on it?
4.
4. So 4 times. And then what is the magnification for the ocular, or the eyepieces?
10.
10. So when you're using the 4 times with this, the 4 times objective, what's the total magnification?
40.
40. Very good. All right, so a couple other points, and then we'll go around and help make sure you're able to use everything properly. This is the condenser. So look in this part of the microscope, feel with your hands and look for like a little lever that's right underneath the stage in the front. That is a condenser. That's important to remember to use. That allows, it kind of like condenses the light in or out. And then of course the turn on switch. Did everybody get that one? And how to plug it in. There's lots of things to think about here. Gotta plug your microscope in, turn it on. And if you turn it all the way on, that's highest light. But then you can control that with the condenser. You can control the amount of light that's actually coming through and hitting your specimen, and then hitting the lenses. And So, some of you, let's see, check out this part of your microscope. Some of you, that's where your actual light is. Some of you have, there's two different versions of the microscope in this classroom. Some of you have a version where you can turn this, and some of you don't have that. Right over here, you guys both have it. So let's make sure it's all. Oh, it's not. I don't think it's working, is it?
I can see it.
Oh, there we go. I'm just turning it all the way up so we can make sure. Leave notes turn it down like that. So yeah. So make sure you have it all the way open. If you have that, a lot of you don't have that ability to even control that part. So okay, cool. Next things to point out are the coarse and fine focusing knobs. There we go. Oh. You always want to mess with the coarse focusing knob, the bigger knob on the side, with the lowest magnification. Even find what you're looking for and get it into focus. And then you do the smaller fine tuning knob to get it nice and focused and crisp. All right? Then that's at lowest magnification. And if you're in the right place on the slide where you want to zoom in even further, then you switch your nose piece objective to the next power up. At that point, you shouldn't need to mess with coarse focusing knob. At that point, you just mess with the fine tuning focus, get in focus, and then if you want to even go higher in power, then you switch it to the next up. We can go all the way to what magnification, you guys?
Yep, that number, 1,000.
Yeah, we can do 1,000 times here. 100 times objective times the 10. And that works better if you have some oil to use, which we don't generally use in here, but sometimes you can still get the highest magnification objective to work for you. So with that, that is your resolving power. Okay, enable. PowerPoint is telling me something weird message. Okay, so resolving power is that ability to be able to see two objects as being separate. And it's easy when you look out in space and you see what looks like a bright star, and then if you get a telescope on it, not a magnifying glass, right, definitely different. And you can actually zoom in on that and see that it's actually two things. Once you get up to the higher power into 1,000 times, it gets more challenging to resolve it and get it to focus in clearly. Adding oil into the mix can help with that a little bit. But what you're using is a light microscope, which means light's being used to pass through the specimen, or pass over the specimen. This is called a compound microscope, compound light microscope, to use with microscope slides. Right behind where Sarah and Deanna are sitting, we have what are called dissecting scopes or stereoscopes, like this one. And those ones use light as well, but they use light, down onto a specimen that doesn't have to be in a microscope slide, and you can actually dissect and mess with it, whereas in a microscope slide you can't so much. So we have both those options here in this classroom, and which is ours go up to 1,000 times, but some compound scopes can go up to 1,500, which is pretty cool. You're going to be looking at some pond water today, which is fun because I think it's work for some cool stuff. We don't know what we're going to find sometimes. We have a mix of there's a little pond outside here, and sometimes it's hit or miss with that because of the droughts we get and things going on. So we have some interesting stuff brewing in the greenhouse back there that we have for you. We have some from the pond out here, it's just right out here, and then some from our mix of crazy stuff. And you're gonna get a chance to do a slide of each and look at those and look for some cool critters. But the point is that they can be living. You can see movement, you can see behavior of organisms. If we want to go even closer than 1,000 or 1,500, then we get into using electrons instead of light, passing through or scanning over the surface of an organism. And we can see something quite a bit closer. We're talking 100,000 times, which is crazy, right? It's pretty cool. 100,000 times what we can see with our naked eyes. And I'm sorry, we don't have one of those scopes here. They cost a lot of money. And they have to be in a really special place. And they take up a big space, very large space. But some of the images we're going to be looking at in lecture and in your reading, you're going to see that they are electron micrographs, meaning that they were taken with an electron microscope, either scanning or transmitting. So TEM or SEM. So scanning is where the electrons are scanned over the surface of the specimen. These specimens cannot be they can't move at all. So all these specimens are going to be dead. Benefit of light, you can look at things when they're live. You can see the plant cell, actually stuff moving inside the plant cell. And then the transmission electron microscope, TEM, there's really thin slices that are made, and then the electrons actually pass through the specimen. So this is actually looking at the same organism using all three different kinds of microscopes. So which one do you think this is? This is TEM. This is transmission electron microscope. And this is, anybody recognize this organism? You might from another class.
Euglena?
Yeah, so this is Euglena. It's a type of protist. And it's, some of them photosynthesize, most of them photosynthesize, but some don't. These guys are so interesting, they could potentially become their own kingdom at some point. Euglenoids. And they are flagellated, they have a single flagellum. Here the slice did not slice through the flagellum, so you can't see it. But they have chloroplasts that are similar to the chloroplasts in plants. Anyways, all right, what's this one? What microscope is this one? What's that?
Compound light.
Just like the one you guys are using. So compound light. How about this?
Dissecting.
This one is actually the scanning electron microscope. So I actually don't have a dissecting example here, but it would look like you just see little green things, because the dissecting scopes can usually only get to 30 to 40 times. But yeah, so scanning electron really shows you the surface of process really well. Is it working?
It's like you move it and it'll actually go back.
Let's go ahead and grab a scope from the 1625. I think Chris just took off. Okay. All right, we're gonna grab another microscope. Okay. So this is kind of fun, just thinking about the fact that what we are able to see with the help of microscopes, and getting down to almost like the molecular level with other kind of stuff, the atomic force, and there's other kinds of stuff that we'll talk about when we cover biological molecules next time. All right, so let's go back to our microscope here. And all of you, these are some good rules for lab drawings. I kind of mentioned it a little bit already. Like if it says to do it in color, please do it in color. Always do it with pencil. Always make sure you read the description of what you need to do really well, like if you terms you need to label. And also total magnification when you do your drawings. In the first few labs, I'll give you a little reminder here, total magnification. And that is going to be, this little table here is just for you to fill out to practice just thinking through total magnification. So if your ocular lens power is a certain magnification and your objective, you multiply those together to get the total. And we all know we all have the same ocular lens power, right? It is 10. You're gonna have 10 all the way down there. And then you've got a spot for each of your 4 objective lenses. All right, so go ahead and you'll be filling that out today, and then preparing a wet mount slide of hot water, which I'll show you that in a bit. But first, for this part, and just to practice getting to know your scope, everybody should be looking at something that looks like, whoop. Let me get this, open this really quick. Nope, obviously that's done. You should all be seeing something that looks like this. Right? Yeah. All right, well, this is your field of view. When you look, you shouldn't see something that looks like this, right? That means your eyes, it's not like right, things that look kind of off. So you want to make sure, first off, you are all seeing something round, that's true. But even more than that, you're seeing something round because that's your field of view. And so inside of that, depending on what power you're at, and I think these are pretty big stem sections, so to actually, I don't think you can actually see the whole round stem, you're just probably seeing part of it, am I right? So let's say you're seeing like a part of it there, and you're seeing there's all these cells. Depending on how close you are, this is going to be the actual stem here, and then you've got all these little vascular bundles in it, which are like veins, and they're just kind of spread out throughout the whole stem. That's what it looks like big picture wise, but then when you zoom in, you see that there are all this background is actually cells as well, and you'll see special larger cells and some smaller cells as you zoom into these actual vascular bundles. So the goal here is we're not getting into the nitty-gritty of what all these cells are called and the components that make up a stem at this point. The goal here is just have something cool to look at as you get to know your microscope, okay? So that's why I'm not having you draw the stem, but make sure you practice going through lowest magnification first, get it nice and crisp and in focus, then move, and that'll be, what's the lowest magnification? 40. And then you're going to move up to what? 100. And then you're going to move to 400. And then you should just go ahead and try 1,000. So you can see how in focus you can get it. And so let's just take a couple minutes to make sure you can work through that. If you've already been through it, try it in a different area one more time. And if at this point, if you have any issues, raise your hand and we'll come around and help you out. Also, try out using the condenser and see what happens when you mess with that condenser. If you have too much light, your specimen's going to be washed out and you aren't going to see detail. If you've got not enough light, it's going to be too dark. Which part's not working? Oh, yeah. Okay, good.
It's cool when you like turn it all the way down.
Take this focusing knob and Closer, closer. Oh, maybe our light's too bright. Let's try this. There we go. I think our light is too bright. Also,
It just was. Condenser is way at 100. So it should be.
Bozo.
I mean, it's there, but it's very
Plug a little.
kind of
But the lights
Oh
definitely
yeah, the
still.
lights.
Oh wait, okay, another thing, and I saw, I think, I saw maybe Deanna, somebody helping you guys with cleaning. Sometimes the lenses definitely do get dirty. Where's
This light.
there's another slide set
Light number.
somewhere. Kelly, did you see
That's
another
weird.
slide set somewhere? that
Was
Sarah
that just
had?
working?
Yeah.
There's a slide set over here. Oh, maybe it's right over there.
Can you turn it?
So, for cleaning the lenses, only use this special lens cleaning paper up here, and you don't have to take a whole sheet of it. Just take a little section, and if you think your lenses are pretty dirty, you can get a little bit of this diluted isopropyl alcohol. And you want to just take your objective lenses and you're gonna rub into them, and it's not gonna hurt them. They're gonna be fine. So you'll just kind of, can I use yours as an example really quick? If you guys can all see this, just take a little bit of isopropyl alcohol on the lens cleaning paper and just rub the base. And this one will kind of push in, and that's okay. The higher magnification ones have that little spring action thing happening so that you don't break your slide if you get too close. So you clean those lenses really well. And then for the final thing, you can kind of like fold it over and then clean your, might as well, you know, if you're cleaning your objectives, might as well clean those oculars too. But don't ever go the other way around because the oculars get more dust and you don't want to rub dust into your objectives. So, that's a good thing to do to clean those. And, let me see, we have one more. Maybe this one will work better.
People are running.
Alright, so how are you guys doing? Not seeing a lot of hands raised, so.
Like the light? Yeah. We don't know. Well, maybe you blew it by putting it to the max. Unplug it, plug it back in.
Right, we did.
Yeah, that's great. These have been in cold storage.
Maybe there's.
It's weird seeing my own eyelashes.
Does she look like Miss Kramer to you?
Remember this?
Like just kind of. Like a nicer version.
Yeah, maybe a little. Maybe this cream or slightly more like in this version.
Yeah.
It's
Just
like
like in the way that
93%.
like Linda's sisters, like there's the evil one and then there's the nice one. Like,
It definitely seems you could be like 93% creamer and 7% difference.
yeah.
Yeah, it's in the middle of the one bigger.
Yeah, but it's always good to start low and then.
Like wish to ponder my orb.
No.
No.
My body interferes with the electrical.
Oh, I can't have you ruining my orb.
Oh, wow.
I forgot how hard that is.
So on one of your
Thank you. All right, so I've done some updating to this section of slides, and well, I actually kind of pulled them in to give a little bit of extra background before we jump right into like large molecules, or the biological molecules. So you'll notice this set of slides isn't actually up on our module yet in Canvas, but I will be posting it, okay? Biological molecules slides are posted and I do it in a PDF format, 2 slides per page. Like I said, sometimes I'm not gonna follow exactly, but often they do, and most everything's there. So, so sometimes I might throw in a couple extra slides here or there, or take something out, but. So this first set, like I said, I would, if taking notes helps you stay focused, I strongly recommend taking notes. But like I said, don't feel like you have to write every little thing down that comes up on the slide or that I say. Just try to write down, major points being covered and whatnot. All right. So, I know what I wanted to do first off. Are you ready for the name test? Are you two gonna help me over there? Let's see if anybody remembers their names over there.
My name's Sarah.
Oh, you're Sarah. Right. How about? Okay, so Sarah gave away her name, and then I added to give away.
I had a class with her last semester.
Kaylee, good job. So Sarah and Haley.
And Deanna.
Deanna will be here a little later. She has a class until like about halfway through. All right. So please don't feel bad if we're still trying to work on names. This is a lot of names to learn quickly. But these cards help. Our introductions help. So. Let's see here. Help me out here. All right. I'm just mixing them up a little. And some of us actually reviewed them after class. It helped. Let's finish. We were reviewing them. So we've got. Yeah. Okay. Yeah. You guys are helping me out, right? Yeah. So say the name and then walk over. Okay, Dylan. Awesome. Okay, this is helping us get those names. The rest of the class, right? This is Dylan. Mm-hmm. And then, actually, Grace. Okay. Eucalyptus.
I'm remembering
All right,
people
what's the
with
best
plans
way?
I like.
I know that does help. Let's see, and then people are sitting in a little bit different places. I remember Grace was sitting there, right? Okay, so that's, sometimes the location helps. I was often, not always, often one of those students that mix things up. You know how you go to class and you like sit in that class, that seat, that first day of class, and then every time you come to class, you go to that same seat, right? It just becomes your spot. And I like to mix things up. I didn't like to sit in the same place all the time. But sometimes that would, I think begrudge other students. They'd be like, wait, that's my seat. I'm like, it's not your seat. It's just, you know. Anyway, so I think it's good if you're mixing it up too. That does make it harder for me to learn names, but that's okay. So, let's see, Sirion, don't say, you guys might be sitting somewhere different. You are! There you go. So go ahead and please put your cards up again. And let's see. I know Destiny. We sit somewhere different today. Let's see. I don't see Destiny here. Hope you are not here yet. Let's see. Oh my gosh, this is trickier than I thought. You guys remember? Abraham. Yeah, Abraham. Are you Abraham? Yes? Okay. Yes. And okay, I remember. Yeah. All right, got Acacia, back corner. You should. I did. Cheater. Were you asking them their name before class because you knew her name? All right. I don't know what this is. That's right, Ben. Okay. And all right, I know this. Yeah.
Caitlin, I can't remember what I can remember.
I thought that was, wait, hold on. Hold on. This one. Casey. Help us remember. Casey, yes. You were wearing a hat last time. I think, yeah. Yep.
Okay,
My brain was
who
thinking
is the
hat.
author
Ooh,
of
I
this?
love that. There's a lot of
The artist
ones I love
responsible for
in
this
here.
beautiful drawing.
All right. Joshua.
Which one is Joshua?
Jacket. Zip up jacket.
That's how you get them to remember.
Zip up jacket.
Okay.
Joshua.
I knew it was over in this group. Area.
All right. And then we got
I don't need this anymore.
Leo is wearing an awesome shirt today. Hi, Leo. And
What do you have to close?
Athena. Okay.
Maybe also nothing put for this picture, right?
Heather. Heather.
You have 10, right?
Yes, Heather. Okay.
Yeah, no, I see that number.
Heather.
Classic.
And
That's what I'm gonna do. I can drop the thing.
Becca. Yes.
That's not opinions on what's going on right now.
It's a little one.
Is your band here today? Oh, there you are. We should ask you. Yeah. And we've got. Let's see here.
Maybe change it to Q.
Right, before. Can you all help us remember Isaac? I think Isaac's here. Man, I hope it didn't
Like
scare.
here was spider and it's almost like a beehive.
All right.
Branches.
And
Like
let's
the
see.
old organizations like that.
Diana, there you are.
Oh, that's how you spell it. Is it Miami or Leon? It's Miami.
That's why. You know what'll help me remember is if I think of Miami. I went to school at Miami University of Ohio, not Miami, Florida. And so, but Miami University, they were formed in 1809, and they were actually a school, Miami University, before, well, Florida was still owned by Spain. So there's like a t-shirt that says that. People are always like, it's Miami in Florida? No, it's in Ohio. For the I think the Miami Native American group that was in that area, where the name comes from. Interesting, huh? So that'll help you remember. I might share these crazy things with you all, and then you're like, what is she talking about? But maybe it'll help you remember too. Miami. Now I wanted to call you Miami. That's what I did to myself. Okay.
Instead of Annie, it's Anna.
Abigail. Abigail? Yes? Go. Okay. And let's see. Kirito? Yes? I see a lot of you with your textbooks. That's great. You don't always need them in class, but then the days you don't have them are like the days you wish you had them, because they have some great images in them when we get into anatomy more deeply into anatomy next week. We got Maverick. Yes? You were looking at me in a way where it was like, nope, I'm not Maverick. But I was like, wait, what's going on here? All right. This lady. You know this lady.
Oh, I know this.
Yes.
JD.
Cool hand in her backpack. JD. And then we've got Sharon with the jellyfish. Okay, right here. Yes. Yeah, Western. What's that? That was the Wednesday. Oh, yeah. We were having a discussion about fungi days. What did we come up with?
We have Wednesday, Western Jack Lantern Wednesday, and Turkey Tail Tuesday, and Truffle Thursday.
I know there was a Toadstool Tuesday, but we really love Turkey Tail.
No, that's really good.
But you can go with Toadstool Tuesday. And let's see, Rita?
We're all Monday.
I said it right this time, yay! All right, and then we've got Jess, very back corner. Very back corner. There you are. Trying to hide back there. Not gonna work. Not gonna work. And then we got Matthew. Yes? Okay. I was waiting for some sort of confirmation. And Elijah. Helena. Yes? Helena. And we're almost done, for reals, because we have a few people who aren't oh yes, okay. All right, we got Vivian, and there you go. I love your flower drawing. And Lindsay.
I like that the roots are visible in the flower drawing.
And Sage with musical notes. What choices to draw? And Oasis. There you go. Love the flag. And then we've got Sam. I'm saying Sam? Yeah? Okay. Well, do I need to say it now? Let me, yeah. Woo! It's warm in here. And then, I'm impressed. You did really well. And there's Sarah, and if you want to make one, you can. We have an old one from Sarah from before in Deanna. So,
It's a lot of tubes.
you can wear it. We can pin it to your shirt. All right. Okay. Sorry it took a little bit of time, but it's helpful with the names. And
Can I also make a quick announcement?
Oh, yeah,
about
definitely.
the-
Hold on one sec. She's gonna be here, okay, great. And she's gonna be, we'll go ahead and put her place saver here.
What's your like?
Don't,
late
nobody
policy?
else should try sitting there. What's that?
What's your late policy again?
You're out of here if you're late. No. It's really important to be here on time, obviously, out of respect for the class. But we're human, things happen, so I really hope people are, being late once in a while happens, but I really hope people aren't continually late.
I feel like you covered so much information and you are introducing a topic that you're lecturing about at the beginning of class, so when you start lecturing, you know what I mean? So
Yeah, and it's really
you miss out
distracting when
on
people come in, late too, and you miss out and stuff, so just try your best not to be late. But like I said, when things happen. Go ahead.
Okay, so a little announcement about the hours that I set for embedded tutoring. For class on Tuesdays, it's going to be 11 to 12:30, and we have class at 1. And that's just because they can't, they have a shift longer than 5 hours, it's only 30 minutes. So if you do need a little bit of help before class, just make sure it fits in that window of 11 and 12:30 on Tuesdays. That's all.
Haley, what should we call that block of time? I still do never anymore.
Yeah, we did.
Tutor time? Tutor hours. Okay, so those are tutor hours and that's on Tuesdays. Okay, and then I try my best to be here about an hour early on Thursdays. If anybody needs anything. That's also time I'm juggling, like, getting things ready too, but I'm here in this classroom if you need to talk or anything. And then, of course, Tuesday nights for a couple hours after class, office hours, open lab kind of time. If you need extra time to catch up on things or ask questions. And I think, and then Kaylee will update us on her schedule in the TRC as well, once she gets that figured out. Or do you have that figured out?
Actually,
If you do, go ahead and, so that's, let's specify this is
in this room.
in 1624. And then go ahead and put, we'll say in TRC, or sorry, I don't know what I'm doing TRC. That's some other thing now on campus that, another different thing going on.
What is it?
It has to do with tenure. It's tenure review committee. So that's my big thing. Luckily, I'm over that. But I do get placed on lots of tenure committee review committees,
So I'm also available on Fridays in the TLC 700s building. It's this way on campus. Does anyone not know where that is? 700 building. Okay, so it's familiar. And I'll be there on Fridays from 10 to 2. at least. 700 one.
Yeah, you just walk in there and you just walk right in the main doors, through another set of doors, and then you're in this humongous space.
It's got a bunch of computers in it.
Yeah, and then you keep going around to the back corner, and that's kind of more the science area. And then you look for Kaylee. And you can go in, they have other hours. Maybe, if you don't mind, like, you could put
TLC hours in
general TLC, if you need to look it up or whatever, general TLC hours. You can go in whenever, and do you know if there's another tutor in there who's taken botany? That you're aware of? There might be another botany tutor in there, but that specific time when you can find Kaylee in there. So, all right, cool. Let's jump into basic chemistry. How many people are taking chemistry this semester? Wow, nice. Well, we're not going to get into it too deep, just at a basic level so we can make sense of biological molecules, and then once we get into more of the physiology part of the semester and talk about cellular respiration and photosynthesis, those chemical reactions can make more sense to you. All right, So take any biological system apart, and we eventually end up at the chemical level. And specifically here, when we're talking like, we can take this even further, you know, into the universe and space, and then our biosphere, planet Earth, ecosystem, community, populations, organism, organ system, organ, tissue, cell, and then the molecules that make up the components of the cell. And here specifically is focusing on a cell from the zebra, heart muscle cell, and specifically looking at a DNA molecule within the nucleus of that cell, and then those components, one of which is oxygen atoms. So we can break it all the way down to that point. So we're kind of starting at this class at that level, and then we'll move up biological molecules, and then into plant cells, and then into plant tissues, and then into plant organs, and talk about physiology and major plant groups. Okay. So this is very elemental, good element, and in what's around us, and we've got matter, anything that's occupying space and has mass, and we've got the 4 physical states of matter. And actually, I forgot, this particular slide is a little older, and plasma is not really considered new anymore, but solid, liquid, gas, plasma. A lot of folks don't tend to think of plasma as that way, but it kind of falls into some of these other categories, so it has its own category. And matter then is composed of chemical elements, and specifically such as carbon and hydrogen, and specifically elements can't be broken down further. You can mess with them a bit, we'll talk about that a little bit, but we have 92 naturally occurring elements on Earth. So meaning if we're saying 92 naturally occurring, what does that mean? Think there might be some that have been produced, but 92 naturally occurring elements on Earth that we're aware of, and we have them listed in a periodic table. So thinking of, and I'll pull up the periodic table again in just a bit, but thinking about living things, because that's what we're focusing in on, and the chemistry classes, organic chemistry, basic chemistry, those are all housed over in the physical science building, which is 1400, right? And this is the biological science building. But, we're all everything's made of atoms. So what are the main 4 atoms that make up living things? Yes?
Aren't they hydrogen, carbon, oxygen, and nitrogen?
Yeah, exactly. So, so those are the ones that make up, the largest portions of living organisms. If we just look at a cell, it would be that way. And looking at humans, our bodies, this is how it plays out. And this is with water in the mix as well. So with that, oxygen then is the largest component of our bodies is carbon. And then you can see here, oxygen, then carbon. If we take the water out of the picture, then carbon is. And then we have hydrogen and nitrogen. But really, you can see because of water, our bodies are quite a bit made up of water, around 80% water, and sometimes a little less. Some plants higher into the 90s, some other organisms down into 60s, 70s% water. And if we look at on Earth's crust, we have, what's the largest component? Is also oxygen, which is interesting. in here, like with living things, like I just mentioned. And then we've got silicon, and then aluminum, iron, and calcium are the major components. So living things are different. And then if we look into, the major elements in space, or I think is helium, hydrogen, yeah, and so it's a different composition of things. If we take the water out of the picture with living organisms, oxygen still, of course, makes up a huge part of living organisms, whether or not they're unicellular or multicellular or colonial. But so you see here when water's removed, so this is the approximate proportion by dry mass of chemical elements found in human cells, and it's fairly similar to other cells of other organisms. And we see that the oxygen changes quite a bit. And what about the hydrogen? Doesn't change that much, but what's water made of? Oxygen and hydrogen, right? But taking the water out, there's still a lot of hydrogen in our bodies. When we take water out, so it changes from 9.5% to 9%. Yeah.
Is that by mass or like moles, or number of?
It's by mass.
Oh, okay.
It's by mass, yes. Okay, that's a really good point. If we're talking individual atoms, right, hydrogen, because hydrogen is so small, it doesn't really make, huge.
Mm-hmm.
I mean, it does. It's see here, it's number 3 of those things that make up the most mass within organisms. But yeah, if it was larger, it would definitely outnumber everything else in mass. Oops.
Ouch.
So going back here, really quick. I know. I forgot this. We need to skip that. This is just showing how, yes, we have those major components that we're made up of, and then we also have trace elements such as iodine, iron, copper, which we need, but only in tiny, tiny, tiny amounts. And if we don't have them, we can get sick. We can have illness. We can have issues occur. Things aren't going to function properly. And so this is an example of what can happen with iodine deficiency. We don't want that to happen. So elements can combine to form compounds, such as sodium chloride. But a compound is a substance containing 2 or more elements in a fixed ratio. And that's what we're going to be getting into with biological molecules. So going down into the nitty-gritty of an element, elements consist of a certain kind of atom. And atom is the smallest unit of matter. What's considered the smallest unit of what?
So.
What about viruses? Wait, that's another camera. We'll talk about that later. All right, so remember, elements are substances that cannot be broken down into other substances, but they can't. Certain components of them can change just a bit, which we'll talk about. So of these, what are the atomic particles of an atom? Yeah, this is basic stuff you learned in elementary school, right? So just a quick reminder on what is the nucleus made up of?
Protons.
Protons and neutrons. And then which of the atomic particles have a charge?
Protons, electrons.
Protons have a positive charge, electrons have negative charge, and what about neutrons? Neutral, hence the name. It's helpful. Helps us remember. And then we have the neutrons and the protons in in the nucleus. So if I'm going back here to our periodic table of elements, and if we look, back, if we look here, pulling carbon out, that's going to be a big focus for today. If we look here, we see the number above is the atomic number, which is the number of protons. And that's the number that determines that element. If that number changes, it's a different element. But can the number of electrons or neutrons change?
Yes. Because
Yeah.
it's ions and isotopes.
But the protons will stay the same, right? So, so atomic number is protons, and if it's an uncharged atom, it's going to have if for carbon it's got 6 protons, how many electrons is it going to have? 6, yes. So because remember, protons are positive, electrons are negative charged. So the same number you would have then an uncharged atom. And what about this number below here, the mass number? That's considered the number of protons plus neutrons. So you can figure out the number of neutrons by doing what?
Subtracting the atomic number from the mass number.
Yeah, so it's the sum of the protons and neutrons, so you just subtract the atomic number from the mass number to figure out the number. And this one is what?
6.
6. So carbon's interesting in that it has 6 protons, 6 neutrons, and 6 electrons. Because the electron number, if it's uncharged, the electron number is going to be the same as the atomic number. Everybody with me on that? So hydrogen is a really tiny atom. What are its components?
The proton.
It just 1 proton and 1 electron, no neutrons. So if we look for hydrogen here, you guys find hydrogen?
Yeah.
Oops, let's go to the one that has, that one's kind of not set up properly. All right, so we see hydrogen, it's just 1, atomic number, 1, atomic mass, it's a little bit more than 1. Okay, so it's just 1, so no neutrons, right? And electrons are so tiny, they're not pulled into that atomic mass number. I mean, they are, but it's so minute that it doesn't really change too much, okay? So what's going on with those electrons? The electrons, since the core is protons and neutrons, the electrons are moving around that nucleus, and it's more forms like a cloud of movement. The electrons will hang out in what are called electron shells. And so if you imagine you've got let me go to this one to kind of see a little bit clearer. If this is our nucleus here, okay, and then this is the space, this first shell from the nucleus. You guys who have some chemistry background, how many electrons can hang out in that space? The first shell.
Oh, the first shell is 2.
Only 2. Actually, only 2. So there's just space for 2 electrons.
6 after this one.
I know. You guys are getting ahead of me here. All right. So then we go to that next shell out, or it's a field, electron field. How many electrons can hang out in that next layer out? How many?
It can't be, yeah, I think it's 6.
It's 8. It's actually 8. So yeah, those of you who are getting ahead, you were right. I heard 8 for the first one. So let's see. I want to go to this right here. So yeah, so we've got. I'm just going to kind of set them here to help us. So if, depending on that number, if all these electron shells are full and there's no empty spots, then that atom is not very chemically reactive. It's if there's empty spots, openings here, that atom is then more chemically reactive, and what's going to happen is that it's going to want to have the electrons come in and fill these spaces. Does that make sense to everybody? Yes?
Wouldn't ever want to get rid of some, or?
There are circumstances where that would happen, but generally if it's over 4 electrons, it's going to want to take them.
This is true, yes. And we'll talk about this more, like, it's important to have a basic understanding of this as we get into photosynthesis and what happens there with light reactions. There's a movement of electrons happening, and even just understanding, this, that when there's spaces, the electron kind of holes or spaces here, they're gonna want to be filled. And we have that actually happening in photosystems, in chloroplasts, with the light reactions part of photosynthesis happening. And hydrogens are broken apart, and the electrons from hydrogen are taken, the protons from hydrogen are freed, and all these really cool things happen that are really amazing. All right, so back here. All right, so what? Before you shout out, I want to give everybody a chance. I don't know, our periodic table is kind of small, so let's see if you can tell. Looking at this one, first of all, how many electrons do you see? 2. 2. How many protons? How many neutrons? So what would the atomic number be? What would the atomic mass number be? All right, so let's figure out what this is. Can you guys see?
It's that one.
What's that?
It's helium.
Oh, that was fast. You already you just know it. And that's fine. I really love that about this class. That we're all coming in with different backgrounds. Some you might not know, and some you know. All right. So we see 2 for atomic number, 4 for the atomic mass, helium. All right, cool. And that's really all we get into it. We're not getting deep into the periodic table. We don't really have time in here, but we will in a basic and a general chemistry class. But chemistry 2A, thank you. And so we talked about that some things can change with the neutron number and the electron number. So if something's different with the protons, then it's a totally different element, okay? But if electrons are missing, or an extra electron comes into play, like this one is creating electrons, if you do have, or it's missing electrons, like hydrogen, if hydrogen's missing an electron, it's just a proton, right? It's going to be considered an ion. If something gets an extra electron, it's also going to be an ion. But then you want to look into whether or not it's got an extra electron, then it's going to have what kind of charge?
Negative.
It's going to be a negatively charged ion. If it's missing an electron, it's going to be a, wait, what did I say? Yeah, positively charged. I was like forgetting where I was going with that. All right, so you got that down. That's called an ion. Isotopes, the electron number is as it should be, but the neutron number is different. So oxygen, these are showing different kinds of isotopes of oxygen. But the neutron number, as you can see, the neutron number is different. So let's go over here, and look at oxygen. Can you guys see that from where you're at? I know it's kind of small, but the atomic number for oxygen is 8, and the mass number is?
15.999.
Can you guys see that from that far away? No. 16, yeah, 16. So, We already covered. Did we already talk about this one? We didn't. Maybe we didn't.
Yeah, we didn't.
All right. So if we look here, this one is actually considered number 16, if that's the norm, right? But just to designate it, because there are other kinds of isotopes out there of oxygen, it gets the number 16. But there's 17, 18, and you've heard of different kinds of just being able to follow, for instance, if you have, I guess like you can tag atoms, is what I'm trying to say, in this way and follow them through a system. And scientists will do that to trace sugars that a plant is forming and whether or not they're sharing them with other plants by way of underground. So, which is pretty cool. You can tag something like carbon, and by way of photosynthesis, have it get pulled into a plant, turned into sugar, or photosynthate, and pass through that plant, pass down to the roots by way of fungi, mycorrhizal connections underground, and then into another plant, and you can trace it. You can follow it along. So, really cool stuff there. Yes?
So what's tagging again?
It's actually doing something like this, forcing an extra neutron in it, and then you're able to then trace it as it moves through a system.
Like you can make water heavier by adding neutrons to the hydrogen, but it acts chemically the exact same as water, so you can trace it. Interesting.
Happy water.
Heavy.
Oh,
heavy
you
water.
said happy. I was like, I hadn't heard.
They make heavy water by adding
that makes
hydrogen.
sense. I thought, did you guys hear that? Did you only hear heavy? Okay.
Is it actually way more?
I guess I'm just feeling happy.
Yeah, you double the weight of the hydrogen.
Like, I haven't heard it called happy water.
So normally
Maybe
we're like
maybe
10,
water's
then it goes
happier
12,
than
and that's
extra.
20%.
Definitely heavier. Yeah, that's cool. Thank you for that example. And then, yeah, we can talk about it more later. All right, so the farther an electron is from the nucleus, the greater its energy. And the number of electrons in the outermost shell determines the chemical properties of an atom. Like I was saying, it's going to be dependent on what's happening in this outer shell of electrons, because if there's any electrons out here, then that shell inside of it is full. So, and if there's any spots open here, then it'll be more chemically reactive, like we see here. And so automatically when, I've heard people when they think about oxygen and they think about, it's got the atomic number of 8, and if I was to ask how many spaces are open in its outer shell, a lot of times people are like, none. Because it's an automatic thing almost to forget that inner shell of 2. And you guys, I think that some of us did that when I was asking, what's going on here, and then what's going on the next one out, and some of you said 6, thinking about that combined. I don't know. It's just it's easy to kind of forget how it works out. So oxygen then should have 2 open spots here, which is what I drew up on the board. So of these different atoms, which one seems like it might be the most chemically reactive? The mix?
Nitrogen?
Nitrogen?
Oh wait, no.
Why are you
Carbon.
saying that? What do you guys think? What do you think?
Carbon has the most open spaces.
So what is that?
It's more likely to take or give away electrons. More likely to interact.
And which one's that?
Carbon.
Carbon, yeah, exactly. And that's actually a big part of why carbon is a really special element, because it has those 4 open spots, and it builds the backbone of biological molecules, carbon. And so when you hear about organic chemistry, organic molecules, it's that carbon in there. And you have, of course, as we know, we also, oxygen plays an important role, hydrogen plays an important role as well, but those are then connected to that carbon backbone. And some of them have nitrogen, an important component is nitrogen pulled into it. Yes?
Why doesn't hydrogen have more spots?
Why doesn't what?
Why doesn't hydrogen have more spots for electrons?
So, good question. Well, hydrogen, what makes hydrogen hydrogen is the fact that it's only has one proton. No other element of all the natural occurring elements has one, and unnaturally occurring has one proton.
Just
Other than hydrogen.
positively charged proton only really needs the one negatively charged electron.
Yeah. And it only has the one electron because it has that one, thank you, sir. It has that one proton. And otherwise, if it didn't, if it had an extra electron, then what charge would it have? It would have a negative charge. If it didn't have one, it wouldn't be, it would just, if it had one, it would be a positively charged. If it had an extra one, it would be a negatively charged ion. If it didn't have it, it would be a positively charged ion. So in order for it to be an uncharged atom, it has to have an equal number of protons to electrons, yeah. But yeah, that hydrogen, that's what makes hydrogen hydrogen, is just that, having one proton. Any other questions?
What happens if like an atom were to share their electron? That'd be pretty cool.
Well, what do you think? So when I talk about chemical reactions, did we plan this? I feel like we planned it. I'm just joking. No, but that's a perfect interlude into what we're going to talk about next. That's why I'm like, that's perfect. Good question. When we talk about chemical reactions, what do you guys think of?
A lot of people
Bonds.
think explosions.
Explosions. I know. But you know, my first thing is like I think about explosion. But chemical reactions are happening all the time, and we're not seeing explosions happening. Good thing, right? Because they're happening many chemical reactions happening in our cells right now, and there's no explosions, luckily. But that can happen depending on what's getting put together, what's getting pulled apart, and that's pretty much what a chemical reaction is. What's coming together, what's getting pulled apart. So Joshua, your question was again?
Well, if atoms were to share their electrons to be friends.
Yes, I like that. So if they can, the fact that carbon has these open spots, it's going to want electrons. And so it's going to steal them or share them. And those are two major bonds, chemical bonds that can happen here. Really quick here, chemically unreactive, I mentioned if the outer shell is full of electrons, chemically unreactive. All right, so chemical bonding. And here we have showing water molecules, and they will. Atoms will want to fill in those spaces. And we know with hydrogen and oxygen, looking at hydrogen, looking at oxygen, oxygen has 2 spots open for electrons, and hydrogen has, bless you, remember, hydrogen just has that, well, there's going to be only 2 elements that don't have a second shell. And they are hydrogen and helium, right? We have helium here, hydrogen. Hydrogen just has 1, helium has 2. So hydrogen it wants to fill up that missing spot there. And they come together. Oh, right here. This one doesn't show water. Where's my water one? There it is. And so hydrogen and, there we go, that's what I was looking for. 2 hydrogens will come in because oxygen has 2 spots open and hydrogen has 1 spot, so they will share these electrons. And that's a special kind of chemical bond. It's a sharing bond, but it's unequal sharing. You guys know what that is? Sharing bonds are called covalent bonds, and then when it's unequal sharing, because oxygen really loves those electrons. It's going to pull those electrons more towards it. And then that's going to make it so that the hydrogens will have a slightly positive charge because the electrons are hanging out more over here on the oxygen side of the molecule. And that makes oxygen have a slightly negative charge. So this would be considered a polar covalent bond. Polar meaning opposite charges on opposite ends of this molecule. Slightly positive charge over here, slightly negative charge over here. So remember, covalent is the sharing. And here's some examples of different kinds of covalent bonds happening. Oxygen gas. We just say oxygen. Whenever we're talking about like breathing oxygen, we're talking about two oxygens hooked together. Oxygen gas. Oxygen, it just doesn't hang out like this. It's chemically reactive. It's not going to just be a single oxygen atom like that. So when oxygen is produced by way of photosynthesis, as we'll get into it more, water is broken apart, two hydrogen atoms come apart, and then you're left with a single oxygen atom that quickly hooks up with another oxygen from another water molecule that got split inside the chloroplast. This is happening like crazy right now with photosynthesizing plants. If they're able to do, if light reactions are happening at nighttime, no. When it's dark, no. But when it's light, water molecules are getting split. Oxygen atoms hooking together, like we see here, forming oxygen gas. And because they're sharing a couple electrons, it's considered a double bond. Whereas two hydrogen atoms can come together and form hydrogen gas because hydrogen's not going to be just hanging out there with that spot open. It's going to hook up with something. It's chemically reactive. It'll hook up with another hydrogen. That's a single covalent bond. Question? I thought I saw a hand raised. Did it get answered? Or were you just stretching me? I don't know where it came from. Someone had. All right. So instead of, so they're sharing electrons. And the most basic biological molecule is this smelly guy here, methane. So we've got carbon. Remember, carbon has 4 open spots. Oh, did you have a question?
Yeah, finally found it.
Well, I asked. I was like, does anyone have a question? Because I thought I saw a hand, but then I didn't see it. I think you were like this. And your name card was like blocking it, I think. Anyways, yes.
Where would you find these like lonely hydrogen and oxygen atoms? Not conjoined.
When chemical reactions happen, they automatically hook up with other things.
So you'll like never find them by themselves?
Not that I'm aware of, unless
Not in
someone
any
else has.
natural setting, at least.
Yeah,
they
not
pretty
in
much
natural
always love to
setting.
be together with anything.
Oh yeah, but that's chemical reactions. I think that like the sun is a big chemical reaction.
Yeah, the
Lots of many things happening there, you know.
Diatomic molecules, hydrogen, nitrogen.
Yeah, that's a good question. I mean, chemical reactions are happening. It's happening. Yeah, for like I don't even know the time that hydrogen would be by itself in the sun, but do you know? But maybe for like I don't know a teeny tiny bit of time. This until something breaks it apart or whatever. Question, comment? I think I saw a hand. Hand raised? No? Yes? Okay. All right. The other kind of chemical bond is an ionic bond, and this is the kind of thing where have you guys been in any kind of relationships or friendships where it's like you give, and you don't receive, or you're just on the receiving side, you're not giving. Maybe later, it's like that, like a parent-child. Parents give. They receive, right? They receive love, everything. But then later in life, hopefully, you know, the child then helps take care of the parent as the parent gets older, because that's hoping. But yeah, so ionic bond is where electrons are being taken and because of that, it forces ions to be formed. An electron stolen from one atom is going to make that atom positive or negative if the electron gets taken away from it. Positive, and then the one that took it away becomes negative. That negative and positive charge pulls them together. So that's an ionic bond. And that's what we get with sodium chloride. And so we see here, sodium, how many, or well, tell me by looking at this, how many, or what's the atomic number of sodium? What I'm trying to say here. Guys, count the electrons. We've got 2, yeah, and then plus 2 plus 8, 10 plus 1, 11. So the atomic number of sodium is 11. It just has 1 electron in its outer shell, and that gets taken, pulled away. The one that has, and we were talking about this, you were saying this, right? The one that has more electrons but missing spots, there's a certain point where the chlorine atom is going to be pulling that electron towards it because it has more electrons in the outer shell, and then it's got its missing spot, its open spot. Whereas sodium only has 1. So sodium's kind of weaker out there in that outer shell, and it loses its electron. Then you have that negative positive charge pulling them together. All right. And this, I love this image. It's really cool. This is a picture of a carbon molecule from an atomic force microscope. And what we're actually seeing here is a pentacene molecule where we have carbon rings. And it's able to actually take an image of electron movement, which I think is really cool. So, so we're not actually seeing electrons or protons or neutrons. We're not actually seeing atoms here. We're just seeing that movement of electrons being shared. Does that make sense? So like right here where there's this point, that's actually the carbon atom. There's a carbon atom, as you can see here. Carbon atom, carbon atom, carbon atom. And then these little points out here are what? Right here, right here, right here, right here. Yeah, hydrogen. And carbon and hydrogen are sharing electrons right here. Carbon and carbon are sharing electrons, and so forth. So that's pretty cool, right? Yeah.
Are we gonna get into electronegativity at all? Like, is that something we need for this?
No. And this, I mean, this is just basic, right? I just want you to just understand basic components of atoms and what chemical reactions are, different kinds of chemical bonds that can form, and that's pretty much it. Yes?
Why is there so much more movement on the two ends?
I'm trying to.
Hydrogen's on the two ends, and the others are with carbons are meeting to those quadrants in the middle.
Oh, cool.
So right here, if you look right here, this would be mimicking kind of what that end here looks like. And then here, you've got electrons moving right there, just between these two carbons. And so there's those electrons here, and here, and here, but right out here, you've got those extra hydrogens happening. So a couple different hydrogens there, So it really is interesting. For a second, I was almost seeing, I don't know if you let your eyes go out of focus a little. It's almost like this shadowy shape, just right there.
Like an optical illusion.
Yeah, it's kind of weird. I was like, oh, what's that? It really does stand out how much brighter it is there from the extra electrons. All right, so covalent bonds, and then the polar we talked about. And so thinking about water actually being formed, which is happening inside of all of our cells right now. We have cellular respiration happening. We're breathing in oxygen, breathing out carbon dioxide. That oxygen's heading into our cells, into our mitochondria, and this kind of thing is happening. We do have water being formed. And then photosynthesis is the opposite. Water is being broken up, and oxygen is being produced. But yeah, that oxygen we're pulling in that we're breathing in is gonna become water. So one final bond I'm gonna mention that we'll talk more about later is a hydrogen bond. And that is because water molecules are polar. One side's more positive, one side's more negative. Which is the more negative side?
Oxygen.
Oxygen side. The hydrogen side, the two hydrogen sides, the Mickey Mouse ear side, is more of the positive side, right? So those positives and negatives, those slight, there's going to be these weak bonds forming between those, as you can see here. And that's why when you see like little tiny organisms are able to kind of walk on water, and there's kind of that little, we'll talk about it more later, but water molecules will stick together in this way. It's really cool. And one of the really special properties that make water so special and why water is so linked to life. So remember, chemical reactions cannot create or destroy matter, they just rearrange it. So if we're thinking about photosynthesis here, where we've got carbon dioxide plus water, those get shuffled around, these components of carbon dioxide, components of water get shuffled around, and we have then glucose and oxygen being produced. And the light is sort of like the activation energy is pushing this forward, providing the energy to make this happen. So with this example of forming hydrogen and gas and oxygen gas forming water, these are the reactants, these are the products. So in photosynthesis, this reaction, which option is showing the reactants? A or B?
B.
So the reactants would be B. So these are the reactants, and then these are the products. So we have carbon dioxide being pulled in, and then we have hydrogens and electrons being pulled in to form glucose, and we have water being split. So we have something being broken apart and something else being built. All right, so just really quick, predict what would happen if a fluorine atom and a potassium atom came into contact. What kind of bond would they form?
I have a feeling potassium
What do you
would
guys
get rid
think?
of its extra electrons.
Look at that outer shell of electrons and numbers.
Yeah.
Am I right?
Yeah, that's a.
So first of all, is fluorine, how many openings does it have for electrons?
One.
Has one. How about potassium?
-1.
Seven. Opens.
Has 7. So what do you think is going to happen?
It's gonna lose the one. Give it to the four.
Potassium can like abandon its child.
Yeah, potassium gets that electron taken, and so we have potassium becoming positive or negative?
Positive.
Positive. And then it makes fluorine then
Negative.
a negative ion. And then, so what kind of bond?
Ionic.
Ionic bond. Very good.
Get adopted.
So,
Fluoride.
now look at this. The reactive properties of or chemical behavior of an atom mostly depend on the number of what? The electrons in each electron shell of an atom, the neutrons found in the nucleus, the filled electron shells, the electrons in the outer electron shell.
D.
Yeah, very good. The electrons in the outer electron shell. Good job. All right, any questions? Before we now, so we've got our basic chemistry jumping into bigger molecules. Oh, whoops. I have a couple more questions. Do you want to do more questions? Let's see how much you understand. Yeah, okay. Forgot I had a couple more in there. Water molecules form hydrogen bonds because of what? The water molecule is polar. The oxygen molecule is positively charged. The water molecules form a tetrahedron. The hydrogen atoms are negatively charged.
A.
I'm going with A.
Why doesn't B work?
Because oxygen doesn't actually have a formal charge here. It's only a partial charge.
Exactly.
You can also work.
Well, the oxygen molecule
It's not actually positively charged.
It's not even an oxygen molecule, right? So I don't know, sometimes multiple choice can be really tricky, right? But if, let's say they said instead the oxygen side of the molecule is positively charged, is that correct?
That'd be more or
It's
less.
negatively charged, right?
Oh, yeah.
So, yeah, these questions can be tricky, right? But yeah, the water molecule is polar, meaning it has a negative side and a positive side to it. And because of that, hydrogen bonds form. An atom can be changed into an ion by adding or removing
Electrons.
electrons. Very good. If you change the number of blank, the atom becomes a different element.
Protons.
Protons. Yes. All right. Good. Oh, shoot. One more. Oh, that is awesome. Whoa, where'd it go? Okay, thinking about the photosynthesis. Okay. Earth's oceans are immense. Small floating photosynthesizers called phytoplankton contribute to ocean productivity. As ocean productivity or the rate of photosynthesis goes up, what would you predict would happen to global carbon dioxide levels?
They go
down.
up because we're still producing a ton of CO2.
So if they, if you can see here, if photosynthesis, if the rate of photosynthesis goes up, then what components are going to be needed? More of what components are going to be used
Carbon dioxide.
and assimilated. What's that?
Carbon dioxide and water are
Carbon
needed.
dioxide and water.
So
So what do you guys think?
CO2 levels go down.
They're still going up. We produce too much.
Well,
Assuming everything else is stagnant.
it depends. Yeah, assuming everything else is just staying the same, then CO2 levels would go down. All right. Good job, everybody. We got through that. That was fun. Right? Are you feeling okay about things?
About the
And
tubes that
hopefully
we need to
that
fill with stuff.
was an okay review for you. And we're gonna, a couple of these slides we're gonna zoom through because it might be a little bit of couple of repeats because the other set was I just kind of threw that together to hopefully give us a good foundation before jumping into this. So other than water, cells consist mostly of carbon-based molecules. And this is organic chemistry. We already talked about that. And we also, we didn't really focus too much on carbon yet, but that's going to be the focus now. But we did mention the fact that carbon has atomic number of 6. And so there are 4 electron spots open there. So carbon can share its electrons with other atoms to form up to 4 covalent bonds, and sometimes forming double covalent bonds with other carbon atoms too. So when a carbon attaches to a carbon, another carbon, often double bonds are formed. And if not, then there's hydrogen atoms connected there. If we're just talking about just a general hydrocarbon chain. And some hydrocarbon chains can actually form rings, like in aqueous solutions. So if they're in water, that can happen with some. And so lots of different, endless diversity of carbon skeletons. Anybody taking organic chemistry in here? You've taken it or you currently are?
Yeah. Well,
Do they?
I'll go the second semester of O10.
All right, awesome. I think it's a great class here. Yeah. Evans does an awesome job. Did you guys have to buy your model kits?
It wasn't required, but it was recommended.
Yeah. I remember taking organic chemistry and you're just like, even during like the tests, you're just like playing, you just feel like you're playing with these your fun little toys. You wreck your sets, you know, and coming up with, figuring out the different components there. And so you'll see molecules arranged differently: structural formula, ball stick model, space filling model. And this is the simplest hydrocarbon, which is methane, which is a greenhouse gas that is often overlooked, but it actually thought to be more powerful as a greenhouse gas than carbon dioxide even. And yeah, this can be questionable depending on the study and whatnot, but it does definitely impact. It is impacting what global climate change and what's happening. And one of the big ones is animal agriculture. It's a big producer of methane. And if you eat too much broccoli, you are too. No, I'm just kidding. So when you're thinking about the amount of fuel that's used, if you're thinking about having like the smallest footprint on the environment and what's happening, of course, a diet with a lot of emphasis in meat, thinking about all the resources involved with producing meat, how much plant food and resources go into getting just that steak on your plate compared to how much green agriculture, plant-based agriculture you can get out of it. And I'm not saying, but yeah, so here we have this, the similar kind of amount of food and the carbon footprint comparing them. Yes?
So is this like energy equivalency or just how much strictly fossil fuels are used in the production? So like, are they counting the like everything for the plants used to feed the bees?
Yeah,
or?
this one is specifically focusing on carbon dioxide, not methane, right? But methane's a whole nother aspect of the picture. But yeah, this one has to do with the gases, what is the output from producing it, all that goes into it. So larger hydrocarbons, such as the octane in gasoline, and comparing that with fat molecules that we eat, you've got those carbon chains. So the shape of a molecule is important in the function, and we'll get into that with proteins for sure. But so we have the carbon skeletons, which have hydrogen attached as well, but depending on which organic compound it is, you might have different kinds of atoms connected to that carbon skeleton. So those are considered the functional groups. And in this class, you don't have to memorize all these. I'm just letting you be aware of them. But if you do take an organic chemistry class, you do get into that in a lot of detail. But depending on whether or not we're talking about a fat or a carbohydrate or amino acid, protein, or DNA or RNA, you're going to have different kinds of functional groups. All right, so biological molecules, most of these are considered macromolecules, very large molecules, and we have monomers. So a monomer is easy to think of what that means. Mono, one, mer means part. So a monomer, a single part. And then a polymer, many parts, made up of many monomers, right? So so most of these are considered polymers, made up of many monomers. And how are they hooked together? Hooked together by way of what are called dehydration reactions, where we see here we have these chemical bonds. And remember, chemical reaction is just the breaking or forming of new chemical bonds. And so here with this reaction, we have an OH, oxygen hydrogen group pulled off, and a hydrogen from another monomer pulled off. And now there's going to be open spaces there and those electrons to be filled. And that then, remember, a hydrogen atom isn't going to just hang out in nature like that. It's going to hook up somewhere. That oxygen is going to have an open spot. you have water being formed here. So a dehydration meaning, if you think about water, dehydrating from a reaction, it's coming away and coming away from your reaction. What do you think the opposite is called?
Hydrolysis.
Hydrolysis. So we have hydrolysis, lyse, to break. Lyse means to break, and hydro, water. So break with water. So if you're breaking, remember you can form chemical bonds, break chemical bonds, water coming in, breaking a chemical bond, and that hydrogen and those two hydrogens and the oxygens then becoming components then of those new molecules, okay? So this is pretty basic stuff, but just so you're aware of it, yeah?
I just have a question, like what is the monomer and polymer like?
Good question.
Well, I mean, like I know in cosmetology school the monomer and polymer is what makes the acrylic nails. So that's why I was like trying to figure out how it's related to this.
Yeah, I actually haven't looked into what those components actually are that are used for nails, but I'm guessing they chemically maybe are single parts, and then the polymer is long many parts hooked together in a chain.
Are polymers just carbon chains, or can they be chains of anything?
My understanding is they can be chains of anything.
Okay.
Yeah, they can be chains of anything. Yeah, that's really, these are good questions. So here, this is just showing these green balls here, we can just say are carbon atoms, just thinking about it like that.
There's some big
All right.
carbon atoms.
So now let's get into the major biological molecules. And then we're going to be taking a break here in just a bit. So we've got carbohydrates. And we've got lipids. And we've got proteins. And then nucleic acids. So, we know those major elements that make up living organisms, and we know there's a lot of other smaller components as well. And those major components of living systems come together to form these biological molecules. And because of the way these molecules interact with different tests that scientists have done through the years in actually discovering these different kinds of molecules, they've been arranged into these specific groups: carbohydrates, lipids, proteins, and nucleic acids. Nucleic acids, super important, and DNA, RNA, are nucleic acids. They have special properties that don't make it so they don't fit into these other groups. And they're the most recent biological molecule that was discovered. And we'll get into that, even like the interesting history about how it was discovered, how the structure of DNA was discovered later this semester, okay? So we're just gonna focus in on lab today. We're gonna focus in on testing and understanding carbohydrates, lipids, and proteins, okay? So focus on those. Carbohydrates, obviously, in the last 10 years, there's been a lot of like anti-carb kind of diets and kicks. Carbs are important for our bodies. What do? What do carbohydrates do for us?
Make us feel greatness.
What do they do? What was that?
Make us feel greatness.
They can definitely help us feel good, and they can also help us feel like crap, depending on how much we're eating. So, depending on what's going on here with our body and how much of them we've eaten, right? But so if we think of simple carbs, well, first of all, carbohydrates are a source of energy, right? They're a source of energy. And plants produce them by way of photosynthesis, and they will store them for later use as starch. And starch is a polymer made up of glucose monomers. So you're gonna be hearing me talk about monomers, polymers on its way. So we have smaller sugar molecules like glucose, monosaccharides, disaccharides. Disaccharides would be like sucrose, maltose.
Galactose.
What's that?
Galactose.
Galactose. And lactose. So the simple sugars, the glucose and fructose, for example, are monosaccharides. So it's two saccharides hooked together. Or I'm sorry, one saccharide, sorry. Honey, let me jump here to sugar. Disaccharides. So two monosaccharides hooked together forms a disaccharide. Glucose plus glucose equals maltose, okay? And then we talked about lactose. Glucose plus galactose is lactose. So those are terms that we're familiar with. We're familiar with fructose, especially when we think about high fructose corn syrup. So glucose and fructose, on their own, you might have glucose added to a sports drink. Glucose is gonna be like practically instant energy for your bodies. Your body doesn't have to break it down well, your body's gonna break it down, obviously, in order to gain the energy that's stored in those bonds. So these molecules have a lot of energy stored in those bonds. Those bonds are broken. When those bonds are broken, our cells have a way of using that energy and converting it into another kind of molecule, over to another kind of molecule called ATP, that then our cells can do work with. So we'll talk about that in more detail later this semester. But glucose can just be broken down very easily in our cells to harness the energy stored in those bonds. If you have glucose connected to another glucose, a disaccharide, your body's got to do that extra step and break apart those two monosaccharides that are hooked together forming a disaccharide, right? If you've got those glucose molecules hooked, forming like a starch molecule, your body's going to take a lot longer to break that down and use. So if you're getting ready to run a really short sprint, what kind of fuel are you going to want? A glucose kind of drink where you can access that energy quickly. What if you're going to run like a marathon? Or maybe we just say like a mile, 5 miles, I'm just kidding. 10 miles or something. Yeah, like a big pasta meal with some potatoes thrown in there too. And your body's gonna have that, it's gonna take it longer to break down and help provide you energy longer. All right, honey is pretty cool. It has both glucose and fructose in it, but the glucose and fructose are not connecting together. So if they're connecting together, they actually form sucrose. But they're not connecting together, and they're just kind of free in there. So if you have high fructose corn syrup, you have a higher number of fructose than other things in there, and our body reacts with that a little bit differently. And some people can have issues if they take too much of that in. So monosaccharides. What's really interesting between, if you compare glucose, galactose, and fructose, they all have the same chemical formula. They're made out of 6 carbons, 12 hydrogens, and 6 oxygens. And noticing along here, you guys can all recognize this, read this, what's happening here. Double bond between the carbon and oxygen here. The difference is where that double bond is, if you're comparing glucose and fructose. And our bodies use it a little differently. But they have the same chemical formula. And you can see galactose and glucose are slightly different from each other as well. So I mentioned sometimes we have rings it might be written out as a ring, or you might abbreviate it, where we just know the carbons are on these points and no hydrogens are added in there, just to make it simple, okay? And that's how we're gonna see it from here on out. So here we have a glucose and glucose. What kind of reaction is this?
Hydrolysis.
Water is getting pulled out of the reaction.
Dehydration.
Dehydration. See that water getting dehydrated from the reaction? So glucose plus galactose makes lactose, and we need a special enzyme. The body does extra work to break this down. Some folks don't have as much of the special enzyme. You guys know what that's called? Lactase. And lactase is a special protein that is an enzyme that has a special shape to it for lactose to fit in there and help break those bonds. Some folks don't have it or don't have enough, and so they would be considered lactose intolerant. And those of you out there who are like that, you know what that does to your body. And that's even an interesting evolutionary discussion to have another time. What's happening there? So the most common disaccharide that we are very familiar with, sucrose, glucose and fructose together, and the enzyme is really easy to name, sucrase. Water, hydrolysis. Remember, hydrolysis, breaking with water coming in, and along with the help of this enzyme sucrase, we have sucrose being broken into glucose and fructose. All right, so that's the monosaccharides, disaccharides. And today in lab, you're going to run some different samples through a test that uses a special solution called Benedict's. And copper, there's copper ions involved here. It's beautiful, giving it that beautiful color. And if there's simple sugars in a sample and we heat it up, then it changes color. So you're going to be doing that today in lab and test for that. For polysaccharides, some major polysaccharides, the one we're going to mostly look at in lab today is starch, which plants produce when they have excess glucose that they're producing by way of photosynthesis, and often they'll send it down in their underground portions. And when we commonly think of, when we think of starch, is potatoes and the underground structures that store the leaves, photosynthesis happening, sending the excess glucose down to underground and storing in special cells in the underground portions. Our bodies can break that down. Here's showing starch molecule, just glucose monomers. We're able to break that down. We have special amylase enzymes to break that down. Plants also will pull their glucose molecules together to form what's called cellulose, which we talked about. What's cellulose used for? Cell walls. Very good. And our bodies can't break that down. So in lab today, we're going to do a test for starch. And this is a really simple test. We just use iodine, which is right over here. And you're going to be adding iodine and seeing if it changes from, you guys know what color iodine is normally? Kind of a reddish brown. People don't use it so much for wounds anymore, but yeah, it's a reddish brown color. And when it comes in contact with starch, it changes to a blue, purple to black color. So you'll do a test that way and actually look at potato cells. And they have these special structures that are similar to chloroplasts. They're a special kind of plastid that takes that glucose, stores it as starch inside these special little components inside cells. If we have extra glucose in our systems, one thing we do with it is we'll hook it together and form what's called glycogen. And a lot of that is stored in our liver. But we can break that down and get glucose if we need it. So that's our way of storing it. or we convert it into other things. But cellulose is actually, if we think about what's the most abundant organic molecule on Earth, it is cellulose because of all the plant material on Earth and that component. On land, it's cellulose. If we're talking about within living cells, it's going to be something else. But major component of wood and dietary fiber. We also have, that would be considered insoluble fiber if you guys are into thinking about your fiber content. Hopefully you're getting lots of good fiber. I want you guys to have clean colons. Is that weird? Because then you can be healthier. You have that good fiber, it cleans out the pipes, helps your body absorb more nutrients. So, so fiber is very important, and we use it every day. We make use of that cellulose. It is complex enough that we talked about fungi have special enzymes, they can break it down. Some microbes have special enzymes that can break it down. Can you think of any organisms that can break it down?
Termites.
What's that?
Termites.
Termites, yep. Termites can break it down, but it's because of these microbes in there that are living in their guts. And same thing with cows and other animals that can just live are just vegetarian, like just living off plant material, herbivores. They're able to break it down too because they've got special microbes in their gut.
Yeah, I've seen cows eat like actual birds. It's not pretty.
All right, so lipids. Lipids are hydrophobic. They don't mix with water. And this is a fat. And if we talk about the category of lipids, we've got fats. That's just one of them, but that's often what we think about is fats. There's other things too that we'll talk about. But if we just look at what a fat molecule is, it's got a glycerol molecule, which looks actually more like a type of carbohydrate. And then it's got these long these long carbon chains with hydrogens attached. If hydrogens are taken out of the picture anywhere along this chain, then we have carbons forming double bonds, and it causes there to be a bend. So it changes the shape of that molecule. So if we're talking, let's see here, unsaturated versus saturated fatty acids, if we're talking those molecules and whether or not they have double bonds or not. If there's double bonds anywhere, it's an unsaturated fatty acid. If it's saturated with hydrogens, meaning no double bonds between the carbons, then it is a saturated fatty acid. So if you hear that, unsaturated versus saturated, it has to do with whether or not there's the hydrogens are there. And so you can hydrogenate unsaturated fatty acids, and that makes them solid at room temp. So if you think about like margarine is made of what's normally a liquid oil, like soybean oil. And you force hydrogens into it, you can make it more like butter, more like a saturated fat. And then you saturate it with hydrogens. So, so the test that we're going to do for fats or for lipids is a really simple test where you're just going to be smearing onto a paper bag and seeing if the smear stays once your control your water dries up. So that is a pretty simple test. Why are fats important? They play a lot of really important roles in living organisms, and one of the big ones is energy storage, especially for some organisms like this whale, where they need that thick layer of fat for energy storage and for insulation. All right, there's so many things to talk about. We gotta move along. This is considered a monounsaturated fatty acid, which would be liquid at room temperature. And you think about it, if all these molecules are stacked together, Here's a whole bunch of, this is showing the glycerol part of the fatty, of the fat molecule. And then the carbon chains here. If there are no double bonds, if they are saturated, they're going to be able to pack in more closely together. If you've got any double bonds there, this would actually show them more spread apart and so forth. And then they're liquid. That makes sense, everybody? How it works? Yeah.
So are the cell membranes made of a similar lipid, or are they different?
They're made up of a similar lipid. They have an extra, and this. You guys are just asking amazing questions. It's great. This you just pulled us right into this. So what makes them different? So what we've been talking about, regular fats have a glycerol and then the fatty acid chains, the carbon chains hanging out here. Phospholipid has this phosphate and choline attached to it as well. And that's important. You guys, as we get to know the phospholipid bilayer cell membranes, this part likes water. This part does not like water. And that's something special of all lipids. They don't mix well with water. And but the hydrophilic head of the choline and phosphate helps make it so that cell membranes can be cell membranes. We'll talk more about that later. But yeah, that's in the lipid category. Steroids are also in the lipid category. And that cholesterol is actually a base steroid. We talk about cholesterol being bad. It's actually good. It's important. We just don't want too much of it. But we need that cholesterol to produce all the steroids we need. And then we have synthetic steroids versus natural steroids, and so forth. All right, any questions about lipids? And you know what I'm saying? One of my slides is missing. Okay, hopefully it's on the one online. I'll have to double check. But I have a slide showing waxes. Waxes are also lipids. So if we talk about beeswax, waxes plants produce, our earwax, these are all examples of lipids. And there's also, there's certain vitamins as well that are fat-soluble vitamins. Do you guys know of any of those?
Vitamin C.
Vitamin C is water-soluble. Vitamin C is one of those ones that you can just have as much of it and you'll just pee it out. But there's certain vitamins that it can actually be bad if you get too much, because your body then stores it in your fat. And vitamin A is one of those actually. Vitamin D also. Yeah, so there's certain vitamins where you don't want to take too much of it, because it can bother you. Yes?
What are vitamins?
Certain vitamins that are fat-soluble vitamins are actually also in this lipid category.
Okay.
So there's a lot of things, when we think of lipids often we think of fats, but there's a lot of other things in there too. So there's the steroids, waxes, and certain vitamins they are biological molecules that don't interact well with water.
Wait,
They're afraid of water.
Certain vitamins?
What's that?
So certain vitamins?
Just certain vitamins.
What are vitamins, like definition?
Oh, a vitamin is, they're something that our body doesn't produce on its own, and we need to supplement that, but they're important components that help our cells function properly. And vitamins have many different kinds of roles, but there's molecular compounds that then our body uses. So people will also think of just something like iron also getting kind of under that vitamin category, but they're nutrients that do more for us than just provide energy and building blocks. They actually help make chemical reactions happen. Yes?
I was going to ask if that technically meant that water was a vitamin, since I don't believe we produce water, but
we do produce water.
how much of it?
Not enough.
Okay. So it still needs to be supplemented.
But and actually some vitamins we actually can produce if we're given the right kind of components. So it's not only, there's more to it than just, you know, something that we need to add to our diet. Yeah, definitely.
Sounds like a loose definition.
Yeah. Yeah, it is, because each vitamin, there's an umbrella category for vitamins and they have different kinds of functions. All right, let's go ahead and take our break and have some snacks, if you guys have them. I do actually have something for this sample today, but it's not a big thing. And, but it'll be during our lab. So let's go ahead and meet back up here, and that clock is still going. Let's meet back here at 3, all right? Okay. And we'll jump. Major protein sources from plants.
Legumes.
What's that?
Legumes.
Legumes. Yeah. So, those are actually seeds. The actual bean part is a seed. And then how about with grains? And actually, certain grains have a pretty high amount of protein in them. Wheat is high in protein. So what are proteins made up of? They're made up of amino acids, and there's a common set of 20 kinds of amino acids. And those then can be hooked together, forming amino acid chains that can then be called polypeptides. The amino acids, there's a core component of amino acid, which is carbon, right? It's an organic molecule, carbon-based molecule, and with hydrogens and oxygen. That's nothing different than what we've been seeing so far with lipids and with carbohydrates. But what makes amino acids unique, really unique, is this amino group, the nitrogen with the 2 hydrogens, and that is the base. All amino acids consist of this. What makes different amino acids different from each other are the side groups. And so those side groups can be composed of all kinds of things. And those side groups might interact with water differently. They might potentially have a different kind of charge. And so they would cause that chain of that polypeptide chain to organize interesting on itself and into different kinds of shapes, depending on what's going on with that side group. Acidic versus basic, hydrophobic versus hydrophilic, and that's what makes these 20 different amino acids unique. But they all have this same thing going on here, the amino group and the carboxyl group with the central carbon and hydrogen. Those amino acids put together by way of a dehydration reaction, forming what's called a peptide bond between a carbon and a nitrogen, which is a type of covalent bond. It's a type of covalent bond like we've learned. It just has a specific name because it forms, we have polypeptides forming. It's a special kind of bond between that carbon and nitrogen. So here's a sequence. The sequence of amino acids is considered the primary structure of a protein. That polypeptide chain here. Changing just one of those can have vast effects, potentially. An example, sickle cell anemia, where we have changing out one amino acid in hemoglobin, which is a special protein that helps with iron, or that helps with oxygen, pulling oxygen in. And if we have this issue here, then it changes shape, and the way a protein functions is by way of its shape, and then that causes the entire cell to change shape into more like a funky shape, like a sickle, and they can hook up on each other and cause capillaries and veins to clog up, and really not a good thing. So, so that arrangement of how those amino acids, the order of them, and them linking together, and the primary structure is what's going to make a protein, and whatever kind of protein it's going to be, and then whatever kind of shape it's going to have. Their secondary structure is pleated sheet and helices, the helix, which sometimes hydrogen bonds form. This is all more detail than you guys are gonna need for like an exam, for instance, but just so you understand the shape of a protein is important. Those side groups are involved in what happens here with the shape. That's the secondary structure, and then tertiary structure, and then how they all, those then, the pleated sheets and the alpha helices, how they all interact together. And then quaternary structure would be if you had different actual polypeptides connecting together, forming a very complex protein, which components that are important to photosynthesis, which we'll get into, would be examples of these. Yes?
So the secondary structure, is that the structures of like the side groups of the amino acids, or is that the structure of how the whole chain shapes itself?
It has to do with what's happening in those, with those side groups.
But like those individual structures, are those just how the whole chain shapes itself through the interaction of the side groups, or is that like an individual side group can form a plated sheet?
So I think I'm understanding, so the side group is what makes them different, that they hook together this way. So you have the actual chain, here's the actual chain.
And then the whole chain makes that shape through the interaction of the side groups?
My understanding is this, so this is your amino acid right here.
Mm-hmm.
And they have there's the peptide bonds. Yeah, that's a good question. My understanding was it was how those side groups interact with each other.
Okay.
Does anybody else know? That's what I think is going on.
So like,
But now
are those secondary structures, are those the shapes of the individual side groups hanging off, or are those the shape of the total chain?
So as in secondary structures in that diagram, it's a little bit hard to tell, but you mean like the pleated sheet?
Yeah, the pleated sheet or the helix, is that the total chain making that shape, or is that the side groups making that shape?
Oh, making it helical?
So do the side groups make the chain helical, or are the side groups helical on the chain?
Going back to this, if you look at how the peptide bond is between the carbon and the nitrogen, right? So they're going to be hooked. This nitrogen is going to hook with this carbon. You see that? That's the peptide bond. That is the amino acid chain.
Yeah.
Right there. So then if you see here, these are other parts that are connecting together. The actual, the amino acids are connecting along this line. So there's the carbon, nitrogen, nitrogen, so like
okay,
that.
so the chain goes
and so
that
it's
way.
actually, my understanding is it's the side groups interacting with another over. So I think I understand what you're asking, but.
So then the purple strip would be the chain going down.
Right.
Okay.
Maybe. And so in this one, the reason why it's forming a helix here is because of the way that this nitrogen then, or this hydrogen, sorry, is connecting with that, is interacting with that oxygen there.
Okay.
So there's other but in my understanding is that it's just helix or pleated sheet kind of structure that happens there. And then there's more interactions that happen when you get into the tertiary structure of the folding of then the pleated sheets and the helices folding over on each. It's complicated, but yeah, I think that's a good way. What you said, I think, is a good way to think about it. If I'm understanding what you're asking, is the purple, that part is the amino acid chain,
Okay.
and another one here, and how they're interacting together is causing that shape. And then you can have this, so this folding happening here, and then multiples of those subunits connecting together, making another shape. So there's a really important enzyme that we're going to be getting into called Rubisco that makes the Calvin cycle happen in photosynthesis. It's a fairly large enzyme, and if it's not there, photosynthesis doesn't happen. We don't have life as we know it. So in proteins, different kinds of proteins are enzymes. We have structural proteins, right, and storage proteins, and proteins like that are in our muscles that help with movement. We have a lot of different kinds of motor proteins within our cells as well. This is just a silly way to think about the structure. And maybe this slide will make it easier to understand. Is you've got your primary structure, which is like, you want to say like the strand of yarn, and then they're twisted, multiple pieces kind of twist together to make the thicker yarn, and then those get woven together, and then those separate pieces, the front, the sleeves, the back, get sewn together, making the quaternary structure. So the different structure. If you're into knitting, this is going to be helpful. If you're not, and that's something. All right, so that's pretty much it about proteins. Protein structure, like I said, shape is really important. For enzymes, they have a special activation site. They have a site that has a special shape that a molecule or two molecules will fit into to help make a reaction happen, to help break bonds or form bonds. So enzymes are super important. If their shape changes just slightly, they won't function anymore. And some of the things that can happen to a cellular environment that can impact those the shapes of those enzymes would be like a temperature change. So like high temp can denature a protein. An easy one to look at is frying an egg. You see the egg white is where the protein is in the egg, and it's fried. Denature those proteins, you can never make that go back. That protein now is changed. It's not as it was. It's not going to do what it did. And other things that can change how enzymes function is also not just denaturing them, but maybe like lower temp or higher temp. One of the things you're going to look at today is when you do your protein test, is you're going to add some enzymes to your protein mix. You're going to test for proteins by looking for amino acids and polypeptides. If you have polypeptide, then you've got a chain of amino acids, right? If you put some enzyme in there, a special kind of enzyme, which right here we've got pepsin. This one will help break peptide bonds. So if we add that into our samples, we're going to test for protein, you can see there might be a slight change of color for whether or not those amino acids are forming a chain, a polypeptide chain there, or if there's just individual amino acids. And so adding that enzyme in there, if we add a lot of it, the reaction's going to happen faster. If we don't add as much, it's going to be slower. If we make it cold, things are going to move around more slowly, things will slow down. If we heat it up too much and we denature the enzymes, then they're not going to work at all. So the final biological molecule, nucleic acids, we're going to save that for later. All right, so I originally was telling you guys we were gonna do something together as a class, but I just remembered how big a class we are, and that's not gonna, my plan's not gonna work because that would mean we'd all be coming to one part of the classroom at the same time. When I've had smaller classes, that would work. But you guys are a large class, so what I want to do right now is quickly, I've pointed out a few things already for this lab. I'll point out where everything is so you know, so you're oriented into where things are in the classroom today. But you can always come ask one of us if you get lost where you're supposed to go next or where something is. Sometimes I forget to throw things out that you're supposed to use, so don't be shy about saying, hey, where's this? Or you forgot this, and we'll quickly grab it. But as you can see from your lab, there's a lot of, and actually I forgot to remind you about this on Tuesday. I really wanted you to read through this before coming to class. So I apologize for not reminding you of that, but it's a great habit for you to just get into. If you get a lab, read through it. Read through it before you come to lab, right? To feel more prepared and understand things. That's okay. You don't have time to read all the details, but you're gonna need to read the protocols, the steps. So for instance, this is all really good stuff to read. Good background I put together here. You can read that at home. So for today, I want you to jump into, and a lot of it's stuff we've covered already, but a nice review. I've got some great images here showing some of the different carbohydrates. And then right here, Benedict's test. This gives you the directions for how to run a Benedict's test. And so you're gonna do the microscope part. I know I had you guys all get your microscopes out because I want you to make sure you get a chance to see this. But that's the part I'm changing it. We were gonna do that together as a group initially. But now, remember I said I want you to kind of group up as in groups of 4. You really could have somebody set up that slide and make sure everybody in your group looks at it to save time. When you're doing group work in this class, we don't do it a ton, but it's really nice to work together as a group in the way of somebody can do this part, someone can do this part, but you're all working together and sharing your results and seeing what's happening. So I definitely don't want, if you're doing group work, the benefit of working as a group is you can get things done quicker and you can help each other out, make sense of things. Please don't be that person that's not helping out, right? That's just kind of like waiting for someone to tell you what to write. Be active in your group and be helpful. And also don't be the person that's controlling everything and trying to do everything. And it's okay if someone in the group makes a little mistake, right? Because we're all learning here, and so it's okay to let everybody do something, right? I know, so I'm saying that because I know there's some folks who might be like, I want this to be done perfectly, I'm gonna do it all. And that's not what this is all about. It's all about us learning, and we also learn to work together as a group. Yeah.
Are these labs perhaps graded on completion?
Yes. And why? Why are you asking that?
Well, since if someone makes a mistake, should we like publicly humiliate them or?
Well, no, that is not where I would like this to go. Good point though. These labs are to be definitely completed and done. And if you're in a situation where a mistake occurs, you have to do a test over. These tests, none of these ones really take a ton of time to wait for. Maybe like max, one of them is 5 minutes to wait for results. So you can do it again. It's not a big deal. But if you start to run out of time for the lab, come let me know. I'll work with you. Sometimes we end up rolling a lab over a little bit into the next class period. That's okay too. And the goal is definitely to get it done. There might have been times before where someone messes up so much that, and there's no time to redo it, and that's a learning experience for the class. And we can look at the class data and discuss it. Okay? That's what we're here for. We're not here to do to specifically get publishable results. We're here to learn, right? All right. So Benedict's test for simple sugars. I mentioned the Benedict's solution is right here. The solution we're using, the Bio-Ret, is specifically for proteins. That's on. I have them far away from each other because they're both blue, different shades of blue, and they're labeled. So make sure you don't get those mixed up. And the test tubes. So here are, I'm going to point out where things are. So pay attention. Test tubes are here. So every time you see tube, you're going to need another one of these. There are permanent markers for you to label test tubes, to keep track of them. So that's where the numbers come into play. And then you'll notice there's these little cubby holes in the front of these tables, and they have test tube holders in them. So potentially, I think you, as a group, one of these should suffice. But if you end up needing another, there should be plenty. You just kind of have to hunt around in these. Sometimes there are ones that are for when we use larger test tubes for things. Don't use these big ones. Try to use the ones that are for these. These ones will actually will work. Well, kind of sketchy. If you don't get the bottom of the test tube just right, it may fall out of the bottom. So be very careful. So just be aware of what your options are, and then pick something accordingly. All right. So test tubes, test tube racks, markers, all the different things you're going to be using. Different tests on how you test different things, and just to see, get thinking about where these things come from. I'm trying to pull in as much plant stuff as I can, or plant related in some way or another. And there's some things that you might not think are very plant related, but they actually are in the end. You'll notice for this one you have water. The distilled water is right over there in the back in the container. Yep, right there. And we may need, actually we may need to pull another one in. We'll pull another one in there. And I'll have Sarah, can you get a couple containers of distilled water, please? That will be a lot easier for pipetting. And do you guys remember where the pipettes are? The little disposable pipettes. I pointed them out last time. They're right over here. So you guys are like, this is a good lab, kind of intro lab, getting used to where things are, what's going on. So these are the little disposable pipettes that you're going to be using. And we need probably two or one decent sized container of beaker with distilled water, just labeled distilled water, or DI water. And then you're going to be doing something interesting here where you're taking, making onion juice and making potato juice. So we're going to use mortar and pestle for other things in class this semester. So this gives you a chance to try that out. In order to juice a potato, if you're strong enough, you can grab it and squeeze it super tight. Everybody like, I don't know if I am. I'm just kidding. All right, so what I recommend you do is just take a little little section. We've got a place with a knife here and a place with a knife there. And you're going to cut as small little sections and put a few little sections into mortar and pestle. Add a little bit of that water, DI water, and then you're going to crush it. And crush it till you feel like you've got a pretty good little potato slushy happening there. And then you'll be able to use the pipette to suck out the proper amount of potato juice that you're going to need.
Just 1 milliliter, right?
That's how you're gonna get your potato juice. So look ahead. Yeah, you only need 1 milliliter for this one. And then you're gonna need 2 for the next one. Very good, yeah. So you're gonna need at least 3 milliliters worth of potato juice.
Yum.
Same thing for the onion. Once you've done with the potato, rinse it out, and right here is the only sink that actually I want you to clean these things out in. You're gonna be just quickly rinsing the tubes. When you're done with today's water, you're gonna rinse them and stick them in this used container over here. And then the mortar and pestles, when you're done with those, you rinse them and then set them over here. 2. Dry, and this has a garbage disposal. The other sinks don't, so don't put anything chunky in those other sinks because they get nasty. And be careful. This is a most terrible place for the switch for the garbage disposal, okay? If you drop something down the garbage disposal, do not put your hand on there. Let me know, and I'll take care of it,
Wait,
okay?
that's the switch for garbage disposal?
I didn't even know that switch was there.
Yeah, it needs to be
Covered.
or maybe today I'll tape it down. Because yeah, could you imagine how terrible that'd be? You're trying to get something out of there and your leg bumps it. I mean, seriously.
I feel like I saw a movie with something like that.
What's that? You saw a movie with that?
Like Screamer or
Okay.
something.
All right.
Bro, I gotta be in one name stance to go.
So the iodine test for starch. You are using iodine, and it's a IKI solution. And so for this, you've got this little dropper. You'll just take that out, use your pipette, and get the proper amounts. So today you're going to get used to using those pipettes. We're going to use them for other things too. So when you suck the material in, you'll see on there where 1 milliliter is. And it's right up there at the top, right before you get to the little bowl. And then you let the rest of it suck in, and then you got 1 milliliter. So you're going to do 2 of them obviously for the 2, and then 3 of those for the 3.
So
So yeah, for the iodine one, it might seem a little overwhelming how you're going to set all this up, but what I recommend is that because there's a lot of you, you designate people from your group to do certain things so that there's fewer people out and about. Like if you've got somebody who's going to set up the microscope site, which is what I'm going to talk about next, they're doing that, while somebody else is coming over here and getting the potato juice together, okay? And then once you have the potato juice, take that, figure out how many tubes you need potato juice in, get that figured out, label those pipette, or those tubes, test tubes, and put those in your rack.
Should we start
And
like
keep track of where your tubes are in the rack for which test you're doing. Okay, because you're going to have tubes for the carbohydrate, the Benedict's test, tubes for the iodine for the starch test, and tubes for the biuret for the protein test. That's a lot of test tubes to keep track of. What's that?
could you like label them with a letter and a number, and then it's just easier like
I'll
1,
leave
2, 3.
that. Yeah, that's something that I want you as a group to figure out for sure. If you label with a number and a letter, once you get the solutions in there, you'll be able to know by what they look like, what iodine looks like compared to the Benedict and the biuret. Question?
Are we writing it, like, are we labeling it on a piece of paper, or are we just writing on the test tube?
You writing? Good question. Writing on test tube.
Okay.
Yep. All right. So I think everything is clear here for the Benedicts. Before I continue on with the starch, sorry, I forgot to mention this. Folks, the Benedict's test is the only test you're putting in the hot water. I sometimes test tubes break with this situation, so I have it kind of set away from other stuff close to me so we can monitor things. But once you have your samples, it's best like put all what you've got going on in those test tubes first. Then you add the 2 milliliters of Benedict solution to each one, and you take your tube to mix it. don't ever do this where you put your thumb over it and you're like this. It's not a good thing to do, and you learn that in chemistry, basic chemistry too. Because you never know what you're shaking up is something that's going to hurt your finger, right? So something else you can do is just hold it and tap it here to kind of mix it in that way. And I'll teach you when we do something else where you actually use the pipette to do some mixing. Then of course you gotta get rid of that pipette tip after that. But yeah, you just tap it like that to mix. You get all those ready and for the Benedicts, and then you're gonna take them over here. Carefully make sure this is where, as a group, you're gonna probably want to decide on a symbol to help you remember that's your group, like a star or a flower or something on there as well.
A mustache.
And then you remember where you put it. So you set those in here and you wait 5 minutes. And depending on the color change, as you can see from this test guide, if there's no sugar concentration at all, then it's going to just stay the same color. Slight green, low yellow, hot up to high orange, red, brown. All right? So that's what you're going to be putting down here. The content, the color change, and what you think is the sugar concentration for that. It's important when you do tests to always have what? A control. Awesome. And our control across the board today is going to be water. Yep, the distilled water. Yes?
Sorry if you already covered this, but the protocol for the Benedict's test calls for 5 tubes numbered 1 through 10. How does
Oh
that work?
shoot, I forgot to update that part. Where does it say that? I thought I...
The iodine one also says 5-legged 1, 2, 3.
Dang.
Where does it say that?
Methods for Benedict's test, the first
See,
bullet,
do you see
obtain 5
here?
tests.
I
Methods
updated-
for iodine tests.
Oh,
Yeah.
right there. Do you see how I updated this part? 1 to 10?
Yeah, the iodine test has the same thing.
I was like, oh yeah, I'm updating it. Just, we change things up. I change things up a lot. It keeps things exciting and makes, adding interesting things. So, yes, you definitely want 10.
Should
Not
make
5. Sorry about that. So, yeah, I updated this part and forgot to update that part. So same thing down here.
You already
Looks like it was
tested.
on purpose.
Okay, so right here, same thing. I think it's because I was looking for numbers and 5 was written out as a word, so sorry about that. 1 to 6. All right, I think this is all. Okay, so for the iodine, we already talked about the iodine color change. Okay, so this should be a lot of fun, but it's going to be crazy. And I anticipate being crazy because there's a lot of you. So this will be interesting to see how this all plays out today, how you guys do. So like I said, I initially thought, small group, we all just do this potato slide. It's not going to work, so 1 potato slide per group, unless you really want to make your own, and there's space to make your own, okay? And I would love, we will actually do something similar to this another time, soon. So this is more to focus on the starch part of the potato cell, whereas we'll do something similar where we focus in on organelles of the cells, okay? So for that part, right up here, this is the microscope slide section. And I'm going to take a section of this potato and actually cut it into some other smaller parts so more of you can make slides at the same time. And what you want to do here is using a razor blade. Oh, careful with the razor blades. Don't do what I just did. Remember, we don't want teeter-totters. Honestly, it's gonna teeter-totter a bit, and I'm sorry. So just know that it might teeter a little, because it's really hard to get a fine little tiny slice of potato. And I'm talking like tiny. You don't want it big, or else that cover slip is just not, it's gonna, just wobble. And you're not gonna have enough light coming in through those cells to see things well. So, as the directions say, you got your slide, you've got your little piece of potato right here, right? You're gonna add, I think for this one, because someone else is gonna do it, we'll just have you add a drop of iodine, drop of water, okay, to your slide. The directions have you actually put it under water, in water, fill it up with water, and then paper towel, piece of paper towel right here on one side, drops of iodine on this side, and you wick the iodine in with the paper towel, and this part of the potato starts pulling in the iodine. And you can see where the starch is being stored. Bless you. That's not exactly the best. That's the best way to do it. I think that's how the directions say. So let's do it that way. But and then once you've got some iodine being pulled into these cells, it's ready for the rest of your group to look at. Whoever is in charge of this part, make sure you get a good view of the potato cells, which will look something like this. And a lot of them are kind of overlapping each other, like so. And you'll notice that there are components inside those cells. So these are cell walls. So you know what you're looking at here. Cell wall. And then you're going to have these parts inside of those cells. And if you look really close, you might see some cool little striations happening there. And if they haven't absorbed any iodine, I want you to see how that looks, and make sure you're showing your team something that kind of shows some absorbing the iodine, some not, so they can compare. Okay? All right. The lipids is going to be the easiest test I already mentioned. It's back there in that back corner, the sign for lipids. And there are Q-tips over there. You'll do a drop of water and smear with water on the one of the little paper bags, or paper square. This is from a paper bag. Paper bag square. You'll number them. There's permanent markers over there. There's permanent markers over here. And you'll smear water on one. Smear all the other options there as well. So I forgot to pull out the honeycomb, the beeswax. So I'm going to pull that out, and that is actually what you can sample too. If you want to try some of the honey with the wax and eat that, you're welcome to. And I'm going to have that set over there right by where Sarah is. So you'll use just get a little bit of that with oh my gosh, what are those things called?
Pipette?
Not a Q-tip.
Cotton swabs.
Toothpick.
Oh.
Toothpick, that's how you're going to sample it, eat it. We'll put a trash piece, I cannot speak anymore. Trash container over there for you to drop your used toothpicks in. Get a little bit of that wax, smear the wax on the paper bag, right? What about earwax? How are you guys going to get that?
I just clean my ears. I'm not doing that.
Somebody in your group gets to volunteer some of their earwax.
Sure.
And so you smear some earwax on there. Earwax is cool, right? So many uses. All right. And then everything else you need is over there. The plant oil is coconut oil.
I'm unironically kind of like overwhelmed right now.
All right, final
I am
thing.
also
Oh my
very
gosh, guys. This is all good. Final thing is the BioRad test. Remember, BioRad test, for testing for protein, you do not need to heat that one up. The only one you're heating is Benedict's. This one, you do want to, it doesn't say it in the directions, but in order, when you add the enzyme, when you add your enzyme here, pepsin, to the mix. Actually, that's going to go in. When you add your enzyme here, are gonna wanna wait just a few minutes for those enzymes to potentially break down the polypeptide. If there's amino acids present, are you guys on that page? The protein page. What color are you gonna see?
Periwinkle. No, wait. Yes, periwinkle.
All right, so right now it's this beautiful blue color, like a royal blue color, like your microscope covers. Once you add the components, if there's proteins present, you're going to start looking for a purple color change. If that purple is more of a blue purple, kind of purple, then you've got amino acids present. If it's more of a pinkish kind of purple, polypeptides. And it's tricky to kind of tell the difference. And so I'll have some pieces of paper out over there for you if you want to put your test tubes behind some white paper and have each other help come together as a group and kind of like help each other see. What do you think? What do you think? So there's a couple here where I have you add pepsin to see if that enzyme will work to break those different kinds of polypeptide chains. Okay? We want to see if we can see that happen here. And you might notice this here. This is something these two we haven't, let's see. Albumin, egg protein that is water soluble, and collagen, which a lot of you probably heard a lot about. A lot of people are drinking collagen, and a bean solution. So you just take a little scoop of beans, crush it up, add a little water, easy. That's a really easy thing to do.
No, the beans are right there.
And the beans that we are using are cannellini beans.
What a
They have-
C-tier bean.
They're a light. I tried to find a light colored bean so we could see the color of the violet through. All right, you might want to take just a couple minutes, figure out how you're going to group up. If it's pretty easy to just group up with the people you're sitting by, helps you get to know new folks, or mix it up and wander around if you want. But groups of 4, around 4. If you're like a group of 3 or a group of 5, that's not a huge deal. So try to group up and figure out what things different folks are going to work on, and then you all come together and discuss the results and work through it. All right? I need to get the beeswax. So are you going to remind me?
Can I join your group?
Okay, sure. I'm gonna go ahead and get started on test 1.
I'm gonna do the potato.
Yeah, so I was gonna say, you get to do potato. You're gonna do wax. You want to start getting on test 2?
Wait, wax?
Yeah, wax.
No, I did not read in that one at all.
All right, well, have fun.
I figured one of us could get like all of the stuff for the different tests, so this probably works better, to be honest.
I don't.
So
Probably they're slightly different.
All right, so
How about
if
I
you're
just
the
do the
first
iodine,
one
you do
to
the wax?
sample, you're going to pour it into
gonna do the
the
Benedict
beaker for everybody.
What is she doing?
Don't mix pipettes, please.
Can you do wax? Is it the ear wax?
Okay, I guess you get to do wax now. And you can be iodine.
this is very sad, very unfortunate even.
It's in the back by the water container.
There's 2 10s and then 1 6.
I mean, one thing I can get set up real quick would be the lipid test. Sounds pretty quick. You just set it up and you just wait for it to dry. And the other thing is just the
Get that beeswax out.
Could you
I already want to fill up the test tubes for the protein test with the stuff that they do. Put them in a thing.
Stanford experiment 1, 6 versus 2.
Oh.
She's doing protein, and then
So what you do is you put the water, a drop of water on. And then when you
Nomenclature. Let's just do like 1.2.
1.2.
Or like so I'm doing test 1, so I'll just do like 1.1, 1.2. If you're doing test 2, you'll do 2.1. Like that type
You
of
want
stuff.
something really, really thin.
You cannot see the
And then I have to go get the.
What's the point of a king-sized shark? Can't no candy. 2.1. What do you want our symbol to be, like a mustache?
I don't know. Just like make it something easy. Just like put a horizontal line on it or something. I'll do that.
sometimes in Botony, it just feels like you have access to all this fancy like 10-level lab equipment, but you're like, let me just have this piece of plant or like, let me just like mix sugar and water into a beaker or put flowers into a beaker, and you're like kind of yeah, I'm excited. Like, it doesn't feel as serious as like chemistry labs.
Alright, so the beeswax is over there now. You're welcome to try, sample some of that and eat some of that if you want. And then try to just get the wax part for your actual
Yeah, it's already like
That's all label how I'm
Those are all
how I'm label them.
with the excess
I don't know. I know it's your precious honey. I know that works.
It's kind of dark in here for some reason.
This one, oh yeah. Yeah, remember. I know you kept these for that honey, so I was
Kaylee,
that's all it's on.
can that light be turned on?
Whoa, that's good.
Yeah, that one.
Sorry guys about the light. Please, if we ever do lab and I forget to turn the light on, feel free to go back in the back corner and flip that switch.
I feel so responsible pointing it out.
Did you point that out?
Yeah. 5 points to me.
What's that?
5 points to me, woo.
water? Where do you get the distilled water from?
Over there, in that beaker. Glucose solution.
So like for the first test, 2, 1, 2, 1.
All right. Make sure this is. If there is broken glass, we will all hear it. Oh, wait, did that happen back there? Okay, no. I wasn't talking about you, Sarah. There is a glass right in this corner. There is a place where broken glass can go.
Ooh, already chunky. That place is probably 27, so.
I don't think that counts as
Okay, I got a good drink.
I have no idea.
Over there.
My whole like thing I do for
That's really big. Maybe go to the next corner. But you can take your, that's actually great. A little drink, any corner of it. Yep.
That doesn't look like
You're looking for the potato, there's some actual potato juice right there.
Hold on. Okay, what about 4 then? Honey. Honey's in the microwave. Clear soda.
I'm working on it. What you looking for?
Album?
For you.
6.
Mouth right now.
I guess we like put it on the side and.
Oh my god.
Oh my god.
Oh my god.
Best I got.
I really don't even know what we're looking for. It's a search. I don't know.
I'm gonna shove that in way too far.
When's the last time
Ranging onions is so nice. It feels so good.
Oh, I'm sure. Absolutely.
Especially when it squirts up and gets in your eyes.
Oh,
that
yeah.
feels so nice.
Sure.
Do you want?
Well, I got my
You want
stuff
to
together.
You have glasses. That's cheating.
Ask the teacher if you have
thank
a
you.
or ask doctor or whatever they're called. Starch.
Starch solution.
I like that pen. And
Starch solution over there.
That is great. So
I will do that.
The empty ones?
Hey everyone! I got honey solution and Susan's got sucrose on the way, so.
Sorry guys, see what happens? So many things being pulled out that some things get overlooked. Here's the sucrose.
Okay.
If you want to go ahead and get the honey and sucrose, those are tubes 3 and 4. sucrose and honey. Get 1 milliliter of those.
Is 1 milliliter just a full?
No, it's like just below the bowl. You'll see.
I just want to know if you can get this soy.
I'm looking at it right.
You're in
One drop of iodine. I don't know if that's one drop. Yeah.
Yeah, so, okay. Yeah, I
Is what?
Potato
Okay.
juice.
8 is onion.
Okay. Okay.
8 is onion.
I would recommend to your lab mates to try this first.
Uh-huh. Okay. And then
Like down here in this area, they can kind of move around a little
draw parts of that are just one? Okay. It says draw and describe what you see. Start. Yeah, see if you can find the sage grain around the
Once I start getting a higher power, it's like, because the, you know, it's not a perfectly
Yeah.
it's a really nice light, but kind of throws you off.
Yeah, no, I tried so hard. I'm more used to just chopping up a bunch of potatoes at once to make like a stew or something. I'm so happy. Yeah, I'm so happy right before I went and grabbed food. Yeah.
Cool, looks really good.
Alright.
I'm over to this area where the cells look really good. Are you able to see a cell wall? You actually have to zoom in closer to see it.
Okay. I think I know it on slide.
It's like almost like here.
Yeah, I see that. Alright, and I just gotta draw the cell?
All I need is that water speed.
There's those structures.
Sorry. Oh, sorry. I tried to get out of the way and I got more in the way.
You got it done?
Good. I just got drawn now.
Sarah is a pro.
This is on the 400. So, the very fine lines. I think I might put one on the floor. I can't tell.
Because they're
Oh,
it can be, yeah, it doesn't have to, it doesn't matter what order.
I've seen the grayed out.
But yeah, Sarah's gonna grab a.
so what we're drawing, right, is the very high cells. So just I just had to find the cell wall.
It's very hot.
Thank you for the I'm going to go ahead and start to get the solution, the column going.
May the devil cleanse you.
Benedict. Which ones are you missing? You're not missing. You have all. You have all. You just jumbled them. Okay. Okay. I'm just gonna bring this over there. Double check for me. Is it 1 milliliter or 2?
It is.
I think it's 2. But...
I think it's 2. Yeah, it's 2 to all. Just have to mix.
What's the top with yours? I can get like
That's what I was
Which one soy milk was it 5?
So that was all for the
That's good.
Because when you cut the potatoes, some of so be sure it's for now, you're focusing on the hair, but still, make sense. Hopefully, try to make sense. All right, good. 2, 3.
No, I'll take it.
college life country, yeah.
It's like crazy. It's like kind of bitter.
Everyone else is crushing it for us.
Let me see. And some water.
I think we should do whatever we. Someone does that. Someone does like
Oh, do you just gotta sit there and let it do its thing? See, here's the thing. I was like, I'm gonna put myself up.
Alright,
Potatoes
so
are doing more than
anybody
test.
having a sample? This is
Fucking know
like
how
a
to do
good
more than
sample
test.
size. We're eating. Oh, I did not for testing.
How'd you do that so quick? This onion and potato is actually killing me. Like, it's just so hard to do.
You just go.
I know, but it produces.
Okay, I guess I'm just gonna go put them in hot water.
No, Earl, what? That still tastes good.
Sure. Just grab a few.
Oh, did you guys all see the honey?
Where'd it go? Oh.
Have you started on the slide or should I?
I haven't because I forgot to write down. Because that's
Oh, true.
2 milliliters of soy milk.
Let me take a little bit of that out.
You can tell right there where there's zero wind. Yeah, a little shiny. My thing is keep the
on a cell wall. It just makes it nice to remember.
I just want to know. Where's the glucose solution?
Sorry. Right over here.
That's really nice and thin, but I would probably just take that little portion.
Just take a bit of it? Okay.
Yeah. Did you see it?
I don't want to carry the water.
Yeah, fair. You can do that.